here at UW. We'd really like to welcome you here from all over the country and the world. We're very pleased that to try out something a little bit new for our conference today and tomorrow, where we have keynotes who are wonderful, panel presentations as normal, and also a larger set of flash and poster presentations that we're very excited about. The posters, I think many of you will know, we're just on the other side of this wall that will open later today. So we're very excited about all the networking time that we, in breaks, discussion, and during the posters. So I was meeting with a former PhD student yesterday who's not involved in this conference, and she was asking, who's a junior faculty at another institution, asking about building networks and co-authors, as junior faculty really ask all the time. And I was reflecting on, so she was asking, like, how did you do it many, many years ago when you were a junior? And I told her one of the key, looking backwards, one of the keys was going to a focus conference of this size, where everyone is kind of doing something similar but complementary, but with a lot of complementarities. And when there's instances where there's a lot of networking time, that can be very valuable. So I just invite you all to really use this time in the next day and a half to, get constructive feedback, which this is our purpose on the programming side of what you've done so far. Get help with what challenges you have. I really do think that any question you have, we've got a set of people in the room that will give you help. You can first look to the chairs of the sessions that are our local faculty to provide some initial feedback, and then connect with people that you don't know. So I really welcome you to introduce yourselves to the local people, introduce yourselves to other presenters. Because our goal is to try to provide an informal atmosphere with a lot of breaks to generate conversation. So those are my main points of welcome today, and we're going to move right into our first session, which both shows some local expertise. Karine is a senior faculty member in the Department of Population Health Sciences and a genetic epidemiologist focused on Alzheimer's disease. So just as some keywords, if that is you, if any of those are your interests, you should track her down in the breaks, and then she'll otherwise chair this first exciting session on methods.
Good morning. So the development of genome-wide arrays over the last 2 decades has drastically changed the genetic research of common complex diseases. Results from genome-wide association studies and subsequent meta-analyses of data from multiple studies for a given trait have improved our understanding of health and disease. But there's still room for methodologic improvements, and in this methods section, we'll hear about issues and potential solutions for Mendelian randomization, for imputation, and identifying novel therapeutic targets. So our first speaker, Dr. Tianwan Liu, will present on how gene-environment interactions invalidate genetic instruments in dose-dependent effect characterization. Our second speaker, Madi Mir, will present on the reliability of imputed genetic data for family-based analysis. And our third speaker, Chen-Yang Su, will present on disentangling osteoarthritis-specific genetic effects from obesity to identify novel therapeutic targets. So, Dr. Liu.
Okay, thank you very much for the introduction. So how do I? Okay, thank you. So I'm really excited to be the first speaker of this whole conference. So the session is titled Methods, but as I think of it, it's better to be methods and their flaws. So I don't want to ruin the day of everyone, but I will start by talking about some issues in a very popular method that has been widely used. So, do I use? Okay. So one of the fundamental questions as scientists, and many of us are epidemiologists, to ask is this causal? So we have studied a lot of exposures, a lot of risk factors. We want to understand if any of the associations we detect between the exposure is really causal for the outcome. But the issue, as you all know, is that associations can be confounded. So if you see something significant, that doesn't have to be and always is not a causal relationship. So one of the promises that the study of genetics has given us is the possibility of doing causal inference, or this is called Mendelian randomization. Namely, we use genetic variants as instrumental variables to assess whether the association between an exposure and an outcome is really representing a causal effect. The idea of Mendelian randomization, or MR, is very simple. So if you have this valid instrument, you can just look at the magnitude of association between the instrument and the outcome, as well as the magnitude of association between the instrument and the exposure. So it's just a ratio. That's the most basic fundamental form of a causal effect estimate. Of course, there are much more advanced methods for obtaining the causal effect estimates. However, this comes with a lot of assumptions. So first of all, there's a relevance assumption where the instrument must be robustly and strongly associated with the exposure. So this is a verifiable assumption because we can really test. That's how we identify such instruments. For example, in the genome-wide association study, we can possibly just use the genome-wide significant independent variants as the instruments. The second assumption is independence. So that says the relationship between the instrument and the outcome should not be confounded. So this type of confounding could be, this confounder is simultaneously a confounder of the exposure-outcome relationship, or maybe this confounder is simply confounding the instrument-outcome relationship. And there should not be such confounders. So this is often taken for granted based on the concept of natural randomization. This is why MR is called MR. There are other problems of this assumption, but I'm not going deep into this today. And the last assumption is called the exclusion restriction, where the instrument should not have direct causal effect on the outcome, and any association between the instrument and the outcome should be mediated through the exposure. Okay, so MR has produced some promising findings. Most importantly, we have now MR results predicting now clinical trial results. For example, MR has predicted that there will be no protective effect of HDL against coronary artery disease among a variety of other complex disease. And this is well in line with large randomized control trials, which has cost millions or even more dollars. And another examples include MR saying vitamin D is not causal for many things. And guess what? The large trials, very expensive trials, they all have shown that there's no meaningful health benefit of vitamin D supplementation. And also more recently, our team has been leading and involved in a lot of efforts in using genetics and Mendelian randomization to guide biomarker and drug target discovery. And we will hear one of the talks from our third speaker. But although we have been able to make a lot of progress, there is a fundamental limitation of the traditional Mendelian randomization, which is they're looking at the population averaged effect. Okay, so we take the instruments from an exposure GWAS, we have their associations with the outcome from an outcome GWAS. So these are all averaged based on an entire population. So the beta XY, the causal effect, is a fixed number that you can estimate. However, I think, and I believe many would agree with me, is that an interesting question is are there dose-dependent or nonlinear effects where the beta XY, the causal effect, is actually a function of the exposure? Okay, and there are questions you can ask. So although we know vitamin D supplementation isn't helping in the general population as a whole, but does vitamin D supplementation work for vitamin D deficient patients compared to the normal individuals? Or we're interested in a lot of behavioral factors. Is drinking 1 bottle of beer harmful for people who don't drink? Or if you already drink 20 bottles per week, is that going to be more harmful? So this causal effect may be dependent on the exposure, okay? And I know many of you are interested in other social factors. For example, I'm personally interested in relationship between happiness and wealth. So if I hold some stocks, of course I hope the price can go up. But for some other people, they may have different thoughts, okay? So there have been some efforts trying to figure out how to do such nonlinear MR analysis. A very naive idea is to do stratification. So essentially, we have an exposure, and we just stratify the population based on the levels of this exposure. And then we cut them into groups. Within each group, we can obtain the MR estimate, and that will be the population stratum-specific estimates, and we can look at the trends. However, this has a major problem because when you create such strata, you're effectively conditioning on the exposure. And if you're familiar with the causal diagrams, if you condition on this X over there, you can introduce collider bias because both G, the instrument, and the confounder, they're both causing this exposure under this causal diagram. And if you impose a condition on that, you can create the conditional association between the G and the U. So you can have biased estimates. So one solution that was proposed more than 10 years ago now is to do a two-step stratification. So first of all, we have the exposure and we have the instrument. We just regress the exposure on the instrument and we obtain the residualized X. And then we stratify based on the residuals. And the rationale is like this. So the first step will create a stratification variable because we have regressed X on G. So this creates a variable, let's say R here. So if we condition on this R, this is no longer a shared consequence of instrument and the U, the confounder. So if this works, then we will have a perfect stratification. It's something that that's not causing the collider bias. And people have applied this method to answer very meaningful questions. So for example, there is a very large study a few years ago which used this stratification method to investigate potential dose-dependent effect of vitamin D on a lot of things. This example is non-cancer mortality. So you can see that this has generated a potentially very appealing conclusion. So among people who are vitamin D deficient, we see a very clear protective effect, whereas for people who have sufficient levels of vitamin D, the causal effect estimates, so-called causal effect estimates, are overlapping with the null. However, if you look at the overall estimate, you will notice that this point estimate is outside of any of the stratum-specific estimates. And that's the exact definition of Simpson's paradox, right? Because it says if you observe a trend in subsets of a population, and the trend disappears or reverses in the overall population. That's the exact definition of Simpson's paradox. So with this, there's no way we can make causal interpretations. There must be something going on. And this actually is the reason why this paper, very impactful paper, got retracted. So it turns out that this method has an implicit assumption called homogeneity of G-X association because it's based on a linear regression. But we know that there are sources of effect heterogeneity, for example, due to gene-environment interaction. Okay, so if this gene-environment interaction involves a confounder of the XY association, then effectively, even if we have regressed out the effect of G on the X, this stratification R can still be seen as a consequence of the GU interaction. And again, this comes back to the collider bias. So we have this potential source of bias that may give you, estimates that cannot be interpreted as causal. And we further looked at this causal diagram, and we realized that this environmental factor that is interacting with the G doesn't even have to be a confounder of the XY relationship. It could just be any environmental factor, because if you look at this diagram, it could be another U, which is having some effect on the R. And again, this is giving us some collider bias. And this is really bad because we don't know which environmental factors are really involved in this context. So a simple simulation can very clearly illustrate this. So we just simulated an X, which is influenced by the instrument and the confounder, and Y is just determined by this confounder. And we introduced some gene confounder interaction. So there's no causal effect at all between the X and Y, but you still see this trend saying nonlinearity. Of course, there are follow-up analysis, for example, a non-parametric approach that tries to remove the effect of G on X using some non-parametric methods. However, it has a similar assumption called the rank-preserving assumption, which says that, okay, if you have a counterfactual model and the distribution of individuals with the same instrument value should be consistent with people who have a different instrument value, okay? But this can still be invalid due to the presence of gene-environment interaction, because when you change the value of the instrument, the percentile where this individual would be at in this distribution can be also influenced by potential confounders and the interaction effect. So this assumption will not be held. So this lemma, I would say, has allowed us to do some very interesting falsification tests in real data. For example, just using the UK Biobank data, we know that we can identify instruments for BMI. There may be some gene-environment interactions going on. We don't know what they are, but likely involving age and sex. So we just simulated an X, which is determined by BMI and the confounder, and another Y. So both the exposure X and outcome Y are simulated. So this would allow us, so the G here, the instrument here, satisfies all the other instrumental variable assumptions. It's associated with exposure, it's not confounded, and it's not having any horizontal pleiotropic pathways. But if we run the nonlinear MR analysis, it still gives us some spurious nonlinear trends. And we repeated this for several other exposures. We just simulated the outcomes with none of them having any causal effects, but we still see a lot of spurious nonlinear effects. So just to summarize, so this type of analysis is very appealing. It's answering very important questions, but it requires a lot stronger assumptions compared to the non-regular linear MR analysis. So the traditional residualized method would require a homogeneity of GX association, whereas the gene-environment interaction can lead to violation of this assumption, even if the environmental factor is not a confounder. So what we can do, so we can develop falsification tests. So we have done this in these papers, and we can specifically test for GE interaction for measured E. We can have tests for homogeneity of variance. We can have the simulation, etc. There are a lot of ongoing work we're involved in. And if we can really identify this environmental factor, so we can potentially do stratified analysis, or we can directly adjust for their effects. And finally, it's really important to be super cautious in interpreting such results, and we have been advocating for the triangulation of evidence. So yeah, I would really like to thank my colleagues. Gus Hamilton has done a lot of the simulation work. George David Smith, who first pointed out there's issue with this nonlinear MR analysis. Steve Burgess for a lot of helpful information and discussion. And also thank you for other colleagues, and thank you very much.
We have time for one question.
Yeah, this is very interesting. So just a quick question. If I'm trying to restrict my analysis within the family, so say I'm only using the variation between the siblings and control the family backgrounds, then should I less worry about this nonlinear residualization method? Because in that case, I will feel like there will be less concern that the co-founders will be correlated with your genes.
Right, so that's a very relevant question. So first of all, applying the MR analysis into a family setting is very ideal, although you will need a lot more data to increase the statistical power. That could effectively control for the potential confounders because that is one major source of confounding, of violation of the independence assumption. However, if you're not looking at nonlinear effects, that's totally fine. If you want to study some nonlinear effects, all these these hidden assumptions should still be something you need to very carefully examine. Yeah. Thank you. Thank you.
And I guess if there was just one short question, if not, we can move on. We have 1 minute left. Okay, let's just thank our speaker again, and we'll move to the next speaker.
Hi everyone, good morning. Today I'll talk about the reliability of imputed genotypes data for family-based analysis. We are concerned that the low-quality imputed genotypes may not be suitable for family-based analysis. In fact, family-based and SIP-GWASs are designed to use Mendelian inheritance as a clean natural experiment to obtain unbiased estimates. But if we have problem with imputed data, we may lose that advantage. So that's something we should consider before using that data. Before going forward, I wanted to remind you about the reference-based imputation. Uh, Suppose we have some unobserved genotypes and we have a reference panel. By comparing the sample genotypes with the reference panel, we can guess the probability of having different genotypes in those unobserved genotypes. If we use the most probable genotypes in those unobserved spots, we call it hard calls, and if we use the expected value of the genotypes in those locations, we call it the dosages genotypes. So we basically have two types of imputed data, hard calls, and genotype and dosages. And one thing worth pointing out is that this type of imputation used in UKB data is completely different from the method that Alex Young used in his SNIPER package. The reference-based imputation does not consider the relationship between the individuals, but that type of imputation used the pedigree to do the imputation, and those are these two are two different types of imputation. For the analysis I'm going to show, we used 70,000 SNPs with imputation quality between 0.3 and 1. SNPs have a minor allelial frequency over 1%. We use the white British subsample of the UKB, and that consists of around 90,000 sibling pairs and 4,000 parent offspring pairs. And the sample we used is like the sample used in the Howe SL 2022. One way to assess the imputation quality data is that we can look at the correlation between sibling pairs or parent offspring pairs in all these SNPs we have. In theory, under random mating and under having a Mendelian inheritance, we would expect to see a correlation of half between both parent and offspring pairs. And we looked at that in the data, in the imputed data. We plotted the empirical correlation as a function of imputation quality. Imputation quality is represented by the info score or the x-axis here, and we have the mean genotype correlation on the y-axis. As you can see, for the low quality imputed SNPs, the correlation is well below the expected value of half the dashed line. And the problem is worse for hard calls compared to dosages, and it is worse for parent offspring pairs compared to full siblings pairs. And even for the highest quality info scores, we still have significant deviations from the expected value of half. So we can say we have problems in the data. Another way to test the quality of imputed data is looking at the correlation between sibling pairs, conditional on their IBD states. The IBD states shows how many alleles they share from their parents. If they share 0, their correlation should be 0. If they share 1 allele out of 2, their correlation should be half. And if they share both alleles from their parents, their correlation should be perfectly correlated. So if we look at that for the IBD equal to 2, we have significant deviation, especially for low info score values, and also for the IBD equal to 1 group, and for the IBD equal to 0, we don't want to see any correlation, but we can see correlation, especially for low info score values. So after that, we started analyzing the WGS data. It is recently published by UKB, and it is not like pre-processed yet, and we had to process it ourselves. But we did it. We perform. We use 68 quality imputed SNPs that can be matched with the WGS data with the info score above 92 up to 99, and 46 low quality SNPs, and again the white British subsample, and the SNPs with MAF greater than 1%. So we can ask, what if we perform a simple regression of WGS data, which is the real data, all the genomes are observed and genotyped, so there is no imputation there. So we can, what if we regress WGS data on imputed data, and to see how much was the imputed data representative of the actual data. We did this, and we separated the high quality and low quality SNPs. In this regression, the expected slope should be 1, because we are basically regressing X on X if the imputed data is good enough. But for the high quality, this is the distribution of the resulted slopes. For the high quality group, we can see it's fairly concentrated around 1, but for the low quality group, the peak is at 0, and it's essentially noise, and it's essentially uncorrelated with the actual data. So that's another evidence that we have problems in the imputed data. For the same regression here, we plotted the R squared against the info score reported by UKB. Info score actually should be the R squared from this simple regression, but for the high quality group, it's not bad. It's not ideal, but it's not bad. But for the low quality group, it's except for 1 point, they are all 0. So we can safely say info score is an unreliable metric of imputation quality in the UKB data. Another analysis we did is we looked into the sum SIP difference regression. Theory implies that these two variables, the sum of the siblings genotypes and their difference, should be uncorrelated in theory. So if we do a regression, we would expect to get the slope of 0. Beta here should be 0. We used WGS data on the left for the dependent variable, and we used the imputed data on the right-hand side as the independent variable. If we plot the z statistics for the slope, the normal QQ plot for the z statistics of the slope, under null hypothesis, we should see all the dots to lie on the 45-degree line. But for the low quality imputed data, we can see they are not, and we have we have significant problems here. But for the high quality data, it has better, it's better than the low quality group, but we still have many problems, and we have an asymmetric deviation from the 45-degree line. So that's another evidence that we have the imputed data have problems and can introduce bias in family and SIP-GWASs. We also did the same analysis using only WGS data. We used the WGS data on both left and right-hand side of the regression, and we still have the problem. And although it is the actual data, we are not sure this is because the WGS data is low quality or we have another problem, but we can see many dots doesn't lie on the 45-degree line, and so the Z statistics doesn't have the expected distribution. We did the same analysis using the array data. This is the original data used for imputation by UKB. We used 2,000 SNPs to do that, and we got broadly similar results that I skip and don't show here, but we got the same results. One interesting relationship that we saw using the genotype array data is that we saw a relationship between the MAF value and the level of significance of the slope of the SIP sum and SIP difference regression. We don't have any explanation for that, but we think we should consider this and find it explanation for this. And if anyone has insight on this, we would appreciate hearing that. So in conclusion, the imputed data, especially the reference-based panel, the reference panel-based imputation doesn't have the right properties for family and SIP-GWAS analysis, and we need more stringent quality control for using that data. And for the next step, we are going to extend our analysis using a larger set of SNPs, and we are working to use WGS data and compare it with the imputed data to see downstream effects of using that data, and we are trying to quantify the bias coming from the low quality imputation. And we are interested in developing reference-based imputation methods that consider relationship between individuals and Mendelian lines. Thank you for listening, and we have a post-acquisition in our group working with Alex Young. You may want to consider that. Thank you.
We have time for a couple of questions.
This is great, Madi. I was thinking about the regressions that you ran. You would expect a slope of 1 if there were classical measurement error, right? But the SNPs aren't classical because they're there's 0, 1, 2, and I'm wondering if if you've thought about how kind of the just the nature of the 0, 1, 2 data could be responsible for some of the deviation, some of the slope there. Yeah, that's
I'm not sure.
yeah, something to think about. The other question I had too was, so you have those pictures at the end where you show the whole genome data. Looks a little funny when you do the difference in some regressions, right, in the tails,
Yeah.
where you're deviating from 45-degree more than you might expect. And I'm wondering if have you done some of the sibling correlations with the whole genome data? Like, when you use the whole genome data, are siblings, do they have a correlation of 0.5 reliably?
Actually, no, we didn't do that yet. Yeah, we should do that. Yeah.
That was my question.
Yeah.
Thanks. That was really interesting. Maybe I missed it, but you probably were looking at autosomes, right?
I don't know. Yes.
Okay.
Yeah.
Could your line of thinking be extended in interesting directions because of the peculiarities of how sex chromosomes work? Would that give you kind of more range of different scenarios to look at?
Actually, I don't know. Yeah. Sorry.
Just a quick question about the assumption of random mating versus moving towards relaxing that. Is that quite consequential, unknown? Could you comment on that?
I guess that's important, but our main goal is to just isolate the effect of imputation qualities. We need to consider that, but we are not sure yet how to do that. Yeah.
Okay, let's thank Madi again.
Okay. Great. So, good morning, everyone. I'm very excited and happy to be here, and I really want to thank TAGC for giving me this opportunity to present in front of you today. So I'm Chen Yang Su. I'm a PhD student at McGill University coming from Canada. So today I want to talk to you about osteoarthritis. Osteoarthritis is one of the most common musculoskeletal diseases and the leading cause of disability worldwide. So as we can see across the globe, nearly 600 million individuals have osteoarthritis, and this is projected to increase to 1 billion in 2050. As we can see, when you reach age 40, suddenly, miraculously, you get high prevalence of osteoarthritis, and this increases exponentially as you age over 40 years of age. So this is a debilitating condition, and it's a huge burden on society. So when we look at what are the most common forms of osteoarthritis, we see knee, hand, and hip are generally the most common. And current management strategies for osteoarthritis are focused on exercise or physical therapy, pain medication, and relief. And if you have advanced osteoarthritis, what we can do is undergo surgical treatments, such as a joint replacement. Of course, we don't want to advance to that stage, and we don't want to have to undergo surgery. So is there a possibility we can come up with a treatment for osteoarthritis? For example, developing drug targets that target OA-specific pathways? Well, we can, but drug development is extremely costly and extremely risky. So there's a failure rate of 95%, and it costs nearly $1 billion just to develop a single therapy. So what can we do to mitigate these costs and failure rates? Well, human genetics is one way to tackle this problem. In 2021, the FDA-approved drugs, 2/3 of them actually were supported by human genetics evidence. So that begs the question, can we find drug targets for osteoarthritis using this kind of human genetics approach? But there's one thing we need to consider. So osteoarthritis shows strong genetic correlations with obesity-related traits. Here I'm showing you a genetic correlation across different traits, and in green you're seeing many obesity-related, such as BMI, and different types of obesity and different waist-to-hip ratios and anthropometric traits. And we see that osteoarthritis has strong genetic correlations with these traits. So when we look at osteoarthritis in 2025, we have 962 independent associations, whereas for BMI, we have over 1,000 different loci that have been identified. So this tells us potentially we may have overlapping signals. So maybe there's a genetic basis that's shared between osteoarthritis and BMI. So can we kind of leverage this? But one issue with this is, let's say we want to develop a drug for osteoarthritis. How do we know we're actually targeting OA-specific pathways and not actually just targeting BMI, for example? So that brings us to our question today is, can we find drug targets for osteoarthritis that aren't actually mediated through BMI? Because if we think about it, we don't want a drug that's actually just targeting BMI-related pathways. We want something that's specific to osteoarthritis. So to answer this question, we're using four different analyses integrated together. First, we're using this GWAS by subtraction approach to kind of disentangle the genetic effects of BMI from osteoarthritis. And then we're using a proteome-wide MR and colocalization approach to pinpoint potential proteins that may be causal for these non-BMI mediated osteoarthritis traits. Then we want to perform colocalization to kind of understand functionally how this OA pathophysiology is occurring. And then we want to assess the druggability of these proteins that are causal for osteoarthritis. So the first axis is disentangling these common genetic determinants of osteoarthritis into these BMI-related components and non-BMI-related effects. To do this, we're using 12 osteoarthritis traits across different sites of the body, and we're using BMI from Yango's 2018 study. And we're using this genomic structural equation modeling approach that actually from 2021, there is a new method called GWAS by subtraction that builds on genomic SEM that's able to disentangle these BMI-related effects from these osteoarthritis effects. And what we want to do is create these latent factors of genetic signals, one that's mediated through BMI potentially, and another one that's acting through non-BMI mediated pathways for osteoarthritis. And when we performed this analysis, we saw we're able to disentangle this into two different latent factors. On the left, I'm showing you an OA GWAS signal that's not associated with BMI. And on the right, I'm showing you an OA GWAS signal that is potentially associated with BMI. And as you can see on the left-hand side, once we subtract out these BMI signals, we're getting a lot of attenuated effects and less polygenicity. On the right-hand side, we're seeing this OA signal that's acting through BMI has high polygenicity, as we expect. As a sanity check, we performed genetic correlations across the 12 osteoarthritis traits with BMI. And on the bottom row, I'm showing you the original genetic correlations with BMI and osteoarthritis. Whereas on the top row, I'm showing you BMI with this non-BMI mediated osteoarthritis traits. And what we can see is that the genetic correlation really attenuates. And we're seeing much less genetic correlation with BMI as we subtract out these BMI signals. And this is also what we expect and just tells us that our GWAS by subtraction approach is working properly. Now that we have these kind of non-BMI mediated osteoarthritis signals, can we pinpoint potential proteins that may be causal for these diseases? Because proteins are actually more likely to be or many proteins are druggable and already have drug targets available for them. So here we're using this Mendelian randomization framework that Tianren already discussed. And we want to assess the proteome on these different osteoarthritis traits on this latent factor of non-BMI mediated osteoarthritis. But first, we have to discuss how are we selecting these genetic instruments for this Mendelian randomization analysis. So we wanted to be very strict and very systematic, so we used this kind of strict variant to gene approach where we're selecting genetic variants that are only cis-pQTL, so cis protein quantitative trait loci, for a single protein coding gene. And then we integrate this with variant to gene mapping where the variant must have some biological link to the gene of interest. Just a short primer, a protein quantitative trait loci is just a cis or a protein or a genetic variant that's in close proximity to a protein coding gene. And we're using, we call this cis-acting pQTLs, and they're more likely to have a direct effect on the protein coding gene of interest and thus influence the protein levels. So that kind of mitigates this bias due to horizontal pleiotropy in this Mendelian randomization framework. Another thing we did was we wanted to ensure that this cis-pQTL actually was only a cis-pQTL for a single protein coding gene. So if you have a scenario where you have a variant that's in close proximity to 2 protein coding genes, we wouldn't use this as a genetic instrument because potentially it could be biased due to horizontal pleiotropy. Another thing we did was variant to gene score filtering, where we wanted to ensure that this genetic instrument has strong biological evidence. So we used Open Targets Genetics, which has this variant to gene score. Basically, it uses 4 different modalities of information to kind of aggregate and give a score to each variant and assign it to a gene. And what we want to do is ensure that the variant has the highest score to the protein coding gene of interest. So here I'm showing you an example for a cis-pQTL for the SOS gene. And if we query this cis-pQTL on Open Targets, we see that the highest score is the SOS gene, as we expect. If it's not, we wouldn't use this for our analysis. Great, so now we have our genetic instruments. How are we satisfying this kind of independence and exclusion restriction principles that we need to satisfy? Well, we conducted really stringent analyses. We used different sensitivity tests, a whole bunch of different MR methods, and we also performed colocalization, which actually ensures that we don't have bias. We're trying to mitigate bias due to linkage disequilibrium. So we're using 2 of the latest methods, PW-COCO and SharePro, together in this robust framework. So our analysis, really, we're leveraging 4 proteomics cohorts. These are the 4 largest currently publicly available proteomics cohorts. And we're using these from ERIC, Decode, Finland, and the UK Biobank. And we're assessing testing for causal effects of the proteome on these 12 non-BMI mediated osteoarthritis traits. And we're performing colocalization, again, to mitigate bias due to linkage disequilibrium. And what I can tell you is we pinpointed 27 potentially causal proteins. And what we did was we tried to group these proteins into key molecular axes for osteoarthritis pathogenesis. And we found generally 4 different axes, which I won't go into detail right now. But when we look at these proteins, what are the proteins that we're finding that are implicated in OA? So here's a network plot of proteins causal for different diseases. And in red, I'm showing you proteins with a positive association with osteoarthritis. And in blue, I'm showing you proteins with a negative association with osteoarthritis, where the linkage, so the thickness of the line shows you the number of significant associations because we're using up to 4 proteomics cohorts as exposure. I just want to highlight a few proteins that have strong links to many diseases. So for example, USP8. Here I'm showing you that a standard deviation increase in genetically predicted USP8 may lead to an increased risk of osteoarthritis. Similarly, we're seeing the same thing for HHIP. COL6A2, increased protein level may lead to increased risk of osteoarthritis. And here ITIH1 is interesting, where there's an inverse association where decreased plasma protein levels may lead to increased risk of osteoarthritis. Now that we have these kind of proteins that may be causal for these different diseases, we want to investigate whether or not these genetic variants that we're instrumenting have regulatory evidence within different cell types and tissue types, because that can really give us, help us understand the molecular basis of osteoarthritis. So to answer this question, we're integrating different information from GTEx, which offers many different tissues. And we want to perform this colocalization between these protein quantitative trait loci and these expression quantitative trait loci. And what I'm showing you here is we selected 2 different tissue types, cell-cultured fibroblasts and whole blood, because these may be relevant to osteoarthritis. And what I can tell you is we found 3 proteins, MANIA, USP8, and TNF-SF12, which has strong colocalization evidence in cell-cultured fibroblasts, whereas SPP1, USP8, TNF-SF12 have strong colocalization evidence in whole blood. One of the limitations, though, with GTEx is that we don't really have these bone-specific tissues that we can test. So that's really a limitation. We have broad tissues, but we don't have any bone-specific ones. So what we did was we integrated this osteoclast EQTL data set, since osteoclasts are involved in bone resorption, and we found 4 proteins with strong colocalization evidence within osteoclasts. Great, so now we have kind of proteins that may be causal for diseases. We have a bit of understanding of the molecular basis of these proteins, but our natural question is which of these proteins are druggable? Because if we want to develop a therapy, which ones can we target? So we used the druggable genome from 2017, which is, they basically grouped 4,500 protein coding genes into different tiers of druggability, tier 1 to tier 3. And we saw that 19 out of 27 of these proteins, so 2/3 actually may have a drug, may be druggable. But our question is which of these proteins actually have a drug available that modulates its protein levels? And to do this, we looked at open targets and we queried these proteins, and we found 3 proteins I'm showing you here in red, COL6A2, IL-12B, and TNF-SF11, which already actually have a compound on the market. So they've already completed phase 4 clinical trials, so potentially this could offer repurposing opportunities. So just to summarize and some main takeaways, we're using this GWAS by subtraction framework to strip out obesity genetics and pinpoint a distinct non-BMI mediated GWAS for osteoarthritis across 12 different traits. Using Mendelian randomization colocalization, we're able to pinpoint 27 causal proteins that are implicated in non-BMI mediated osteoarthritis. And 2/3 of these proteins are already druggable, so potentially we may have repurposing options, and the other ones may offer us potential novel therapeutics. So one last thing, if I have a little bit of time, I just want to ask the question of why are these proteins important in a social genomics conference, right? That could be the natural question. Well, this study earlier this year showed that actually these circulating proteins may mediate loneliness and cardiometabolic diseases. So what they showed was that increased subjective loneliness may lead to increased plasma protein levels, leading to increased risk of cardiovascular disease, stroke, and mortality. So how does this relate to our work? Well, previously we conducted this multi-ancestry proteome phenome-wide atlas in 3 ancestries. So we manually curated 355 diseases, and we wanted to test the causal effects of proteins on these diseases. And this is really a collaboration with Satoshi Yoshiji, Tianren Liu here, and Adrian Vandergraaf, as well as the Broad Institute, where we wanted to create this resource, an atlas of proteins causal for diseases. And here I'm just showing you a protein psychiatric trait network plot. So for future work, what we're interested in is we're working with the Broad Institute, and we're leveraging this knowledge portal network, which has nearly 4,000 total traits, so across 1,200 different studies. And what we're trying to do is create across 6 ancestries these proteins that may be causal for all diseases and create this resource for the community. So these are just some references and resources that I used for preparing for this talk. And finally, I want to acknowledge Tianren Liu and Guillaume Butler-Laporte, who were really my mentors for this project, as well as, of course, the TAGC for inviting me here. And I want to also acknowledge my supervisors back at McGill, Satoshi Yoshiji, Siri Zhou, and Vincent Musser, as well as all funding agencies. Thank you very much.
We have time for a question or two.
Thanks. That was a lovely presentation and just spectacularly impressive work. And also, I think, Jason, maybe congratulations are in order for the quality and scope of work that we've seen in just like, I don't know what, the first half hour of this meeting. I've been attending this meeting, or I guess its ancestor, for over a decade, and it's impressive to see how far it's come. Anyway, my question for you is about the early and sort of simpler part of the talk before you get into the proteins, the GWAS by subtraction. You showed us a table of the genetic correlations between your residualized OA constructs and BMI.
Yeah.
And I was surprised to see just the next slide after this one, I think. So this was that the Manhattan plot was super persuasive that the approach was successful in identifying maybe a small number of loci that were influential of OA. But I'm looking at this table, and the residualized correlations
seem higher than I would have expected them to be. And I was wondering if you could just tell us how you did the residualizations. Was it on the latent construct or on each of the individual subcomponents? And what should we expect those residualized genetic correlations to look like?
When you're talking about higher than expected, you're talking about genetic correlation you're expecting maybe something below 0.1 in the top row, or is it, could you clarify?
Let's say the correlations are cut by maybe half, sometimes a little less.
Yeah,
And if I compute a regression residual, the expected correlation should be 0. So
yeah, that's
my
a,
expectation is wrong, but I'm asking for help understanding why.
Yeah, that's a great question. So that's something we did also consider. And I kind of expected this question as well. So essentially, when we're subtracting out these BMI components, we're not actually subtracting out everything. So there's always some residualized components. And we saw this when we were looking at the proteins that were causal for different diseases. We still identified certain proteins that may be causal for example, BMI-related traits. So one of the proteins we found was ASIP, and this actually, previous studies have shown that this protein, when you decrease the levels, you may potentially, it's implicated in monogenic obesity. So we're subtracting out this BMI component, but we're not taking out everything, and there's still some residuals remaining within this framework. But does that answer your question? Okay. Okay. In the back.
Thank you for the talk. I worried that the structural equation model in the GWAS by subtraction could be wrong. And for example, if someone has osteoarthritis and they will have less physical activity and that increases BMI. So when you subtract out this BMI component from OA, is it possible that creates some artifact?
Yeah, so that could be, that really goes into the model itself. So there are different kind of, the real strength of kind of this genomic SEM approach really is the flexibility. So it allows us to test different kind of model assumptions and potential violations of the model. And this kind of flexibility really, one way we could test this is, for example, let's say in this framework, we're assuming the covariance between this GBMI here in blue and G non-BMI, the covariance is 0 between the two. That could be true or that could be false. And one way we can specify this is we can really change these parameter settings within the model and basically rerun this GWAS by subtraction, look at the GWAS we get, look at the novel loci that we're finding, and see if this kind of relates to BMI traits or any kind of osteoarthritis traits, and see if anything changes. One thing we could also do downstream is perform the proteome-wide MR again and see if any of these targets change dramatically. So there are different kind of sensitivity analyses that we can do by altering the model specification and just testing this to see if potentially the GWAS by subtraction has any potential issues.
So just one final question. Thank you for this talk. It was great. I was curious actually at the end of this, when you get to these druggable targets, do you have people that you're working with in order to do sort of, I think like target trial emulation studies in any kind of large data set that has both GWAS and electronic health records would be a way to test if these are actually showing these effects in people that are already taking them, considering their existing drugs. Do you all have collaborators that you've planned to sort of bring this downstream with? And are you also planning on looking at any other diseases? It seems like this pipeline and approach could be applied to any number of sort of important chronic diseases.
Yeah, that's a really great question, and that's actually the most important question that we're interested in is the downstream application of this. So one of the targets that we actually identified from our first study here was this protein IL-1 RL1, which is implicated in inflammatory bowel diseases. And as you know, inflammatory bowel disease, it's an autoimmune disease. There aren't many therapies available. And this protein that we identified actually already has druggable targets undergoing phase 3 clinical trials for asthma. And we're actually collaborating, or we've been in talks with pharma. I can't disclose the company that's developing the drug for IL-1 RL1 therapies for asthma, but we're actually in talks with them to see if potentially we could repurpose this. And the main limitation really is To kind of assess this downstream, you need funding. So definitely you need collaborators within pharma who have kind of deep pockets to perform kind of, we produce the targets and then they can downstream validate these. We do want to, my supervisor does have collaborations with pharma and they're very interested in this, but as a student, I don't have much leverage in this position, so I'm just running the analysis and doing what they tell me to, but of course, as a slave. But downstream, after I finish my PhD, I'm definitely interested in pursuing this further. But yeah, thank you for the question.
Questions for Chen Yang? Approach him during one of the breaks. Let's thank him one more time.
My name is Lauren Schmitz. I'm an associate professor here at the Follette School of Public Affairs. I do work on gene-environment interactions and also biological aging and epigenetics. And so I'm really excited to introduce our first flash session, which is going to highlight some more papers that are looking at biological aging and epigenetics. We'll be hearing from two fabulous guest speakers today and tomorrow on these topics. But let's get started. So I'm going to go ahead and call the first four flash session presenters up here to sit here. You each have 5 minutes. I'll be sitting there with the card. So when you're 10 minutes or when you're at 2 minutes, I'll hold up a sign. And then when it's time, I'll go like this. And then after those four presenters are done, we'll bring up the next four. So if I could have my first four, Kelly, Lauren, Zoe, and Wesley. And Kelly is going to go ahead and start us off. Kelly Renchester, thank you. And we'll hold all questions for the poster session.
Okay. All right. I will kick us off with a talk on, very brief flash talk on lifetime chronic stress exposures, stress hormones, and biological aging in the midlife in the United States study. I'd like to start by acknowledging Jenna Hansen, who is a fantastic postback in my lab, who's now a graduate student at University of Miami and led the analyses. So a variety of psychosocial stress factors and adversity in both childhood and adulthood has been linked to greater biological aging and several different hallmarks, including inflammation, telomere length, and epigenetic alterations. The diagram shown on the right here outlines about a dozen hallmarks of aging that have been identified. However, research on the impact of stress on cellular senescence in particular, which is a key hallmark of biological aging, is less well developed in the literature. However, in animal and in vitro studies, research has shown that exposure to stress hormones, catecholamines and cortisol, increased DNA damage and cellular senescence. However, these associations have not been investigated in humans, and so we sought out to look at these associations. So the Midlife in the United States study is led by Carol Riff here at the UW. This analysis included 531 adults in the Refresher 1 cohort that had complete gene expression data and stress hormone data. The mean age for the sample was 54 years. So I wanted to point out that MIDAS has gene expression data at one time point. And they also, in the last year and a half, added epigenetic aging data. So this is now a fairly large cohort to examine some of these questions. The participants completed the Stressful Life Events Inventory, and this assessed chronic stressors across childhood and adulthood. Jenna actually coded each of the stressors into these categories. There were 17 different events for childhood and 20 for adulthood. The participants completed a 12-hour urine sample, overnight sample, to assess epinephrine, norepinephrine, and cortisol, and then provided a fasting blood draw the following morning that we used to assay RNA sequencing of peripheral blood mononuclear cells, or PBMCs, to assess some of the molecular markers or processes in this cellular senescence pathway. So if you aren't familiar, cellular senescence can be measured using a senescence signal P16-INK4A. The gene that encodes this protein is CDKN2A. The DNA damage response is thought to be upstream of P16 expression. So when cells reach a certain level of DNA damage, it can initiate cellular senescence, and then senescent cells secrete a whole host of pro-inflammatory factors called the senescence-associated secretory phenotype. So we calculated gene composite measures. The DNA damage response is comprised of 30 genes, and the SASP is 57 genes. Please see my poster with our results. There's more results than what I could show here, but I do want to direct your attention to, we conducted a mediation model. These are cross-sectional mediation models, so we really thought about this as exploratory work for future longitudinal work. So I want to say that first. But we did want to examine norepinephrine, epinephrine, and cortisol as statistical mediators and potential mechanistic pathways through which stress would influence these biological aging parameters. And first, what we found, is that childhood stress exposures accounting for stressors that occurred in adulthood were significantly associated with norepinephrine and moderately or marginally associated with epinephrine. We found in the first study in humans that norepinephrine was also significantly associated with the DDR and pro-inflammatory SASP processes. So this suggests that higher circulating basal levels of norepinephrine in midlife are associated with or suggest a higher senescent cell burden in midlife that could predict future onset of chronic illness. It does certainly, we think that it would increase risk. Senescent cells are thought to play an important role in the pathophysiology of age-related disease. And then when we test this mediation model, we found, surprisingly, that childhood stress exposures were not directly related to these biological aging markers. However, there was a significant indirect effect, suggesting that it's really through higher basal levels of norepinephrine in midlife that exposure to stressors in childhood is associated with biological aging. So I'd love to talk with you more in this session. Thank you.
Okay, and next we have Lauren Psasnik.
Good morning, everyone. My name is Lauren Psasnik. I am at Northwestern University, but my work today is going to be on my dissertation at University of Michigan. So today I'm going to be presenting on depressive symptoms partially mediate the relationship between psychosocial factors and epigenetic age acceleration in a multiracial ethnic sample of older adults. So the primary motivation behind this study is that epigenetic age associations with psychosocial factors have been largely uninvestigated, particularly in large multiracial and ethnic populations. So previous research has found that psychosocial factors, including both stress and loneliness, have been linked to epigenetic aging. However, these results have been relatively inconsistent, with other studies reporting no significant relationships. And furthermore, understanding the role of mental health on these relationships can help to guide early intervention. So the aims of this study were to assess the associations of cumulative psychosocial stress, loneliness, and depressive symptoms on epigenetic age acceleration. We also wanted to investigate whether these relationships were modified by sociodemographic factors, including sex and educational attainment. And then finally, we wanted to investigate whether depressive symptoms mediated some of that relationship between psychosocial factors and epigenetic age acceleration. So in order to conduct this study, we used data from about 2,700 participants from the Health and Retirement Study, or HRS. So cumulative stress and loneliness were measured in 2010 or 2012 from the Psychosocial Leave Behind questionnaire. as were participant sociodemographic factors and health behaviors. So this cumulative stress score, I can talk more about it at my poster, but it was created from 6 stress domains across the life course, and loneliness was measured using the UCLA loneliness scale. And then we also assessed depressive symptoms from the CESD as our hypothesized mediator in 2014, and then we finally collected the epigenetic clocks in 2016. So when conducting multivariate linear regression models, adjusting for sex, education, marital status, employment, having children, genetic ancestry, and white blood cell proportions, we found that most second and third generation clocks were associated with stress, loneliness, and depression, with about a 1/3 to a half-year increase in epigenetic age acceleration for 1 standard deviation increase of the psychosocial factor. So we did look across 5 different clocks, ranging from the Horvath first generation up to the pace of aging third generation. And then if you see here in Model 2, we also adjusted for health behaviors, including smoking status, alcohol use, as well as BMI. And these attenuated the associations with some of the clocks, but not others. And most of these associations did remain FDR significant. So we were also interested in assessing psychosocial factor by sociodemographic interactions on this epigenetic aging. So when we looked at whether the relationships differed by sex, on the left here, you can see that stress and loneliness had a much stronger effect on epigenetic age in females, which is shown in blue, as compared to males, which is shown in red. So this was significant for the pace of aging clock, but not the other clocks. And then we also found that loneliness had a much stronger effect on epigenetic age in second and third generation clocks in those with no college degree, as compared to those with a college degree, which is shown here on the right. And so in particular, we found significant interactions in the pheno, age, grim age, and pace clocks. And then finally, we hypothesized that depression could be potentially on the causal pathway between psychosocial factors and epigenetic aging. So when we examined whether depression was mediating some of these effects, we did find that particularly for grim age and pace, depression partially mediated some of those relationships between stress or loneliness and epigenetic aging. And that ranged, depending on the clock and the exposure, anywhere between 25 and 40%. And then although results aren't shown here, after adjusting again for health behaviors, those only slightly attenuated those relationships. So in summary, psychosocial factors, including both cumulative stress and loneliness and depressive symptoms, were associated with epigenetic age acceleration in a multi-ancestry population. Females and those with a college degree were more sensitive to the effects of psychosocial factors on epigenetic aging. And finally, health behaviors may mediate some of the psychosocial factor epigenetic relationship, but not all of it. Mental health, including depressive symptoms, may also play a mediating role on that relationship between psychosocial factors and aging. And I'd quickly like to thank my advisor, Jen Smith, and the rest of my colleagues at Michigan, as well as Lauren Schmitz and the colleagues at Wisconsin for this work. Thank you.
Lauren. Now we will have Zoe Lai.
Okay. Hello, everyone. I'm Zoe Lai, and I am a postdoc research fellow at Wednesday University and the Institute of Gerontology. So today I'm going to talk about a research in which we look at the employment trajectory and biological aging with the focus on racial ethnic disparities in work instability and epigenetic age acceleration. As we may know that employment can be considered as a social determinant of health, which associated with a lot of which associated with mortalities and a bunch of age-related chronic disease. And also, job instability and employment could be a chronic stressor which associated with negative health impact, especially in later life. So However, there's a lot of research gap within the field because most of the research tend to look at the cross-sectional employment data. For example, like they look at employment versus non-employment, but overlook the lifetime trajectory that people can experience over their life course. For example, some people, they may experience like multiple job disruption and experience long-term non-employment, or they enter the labor force later than others. So there's a lot of nuance that we can look into when we come from a life course perspective. And then also, the weathering hypothesis, they also propose that maybe the chronic stress exposure to socioeconomic factors is also associated with the premature aging, especially for Black and Latinx adult. That being said, we can hypothesize that maybe the repeatedly stress exposure to those chronic stressors can actually exacerbate their biological aging in related to their chronological age. So fortunately that we have the epigenetic clock that allowed us to actually provide out information to precisely measure their pace of biological agents. So for these studies, we try to examine that whether there's a physiological wear and tear is associated their stress exposure to job and employment trajectory on their biological aging. So that leads us to our research question in which we look at how employment trajectory, which age from 80s to 50s, is associated with their epigenetic agents, and does those relationships differ by race and ethnicity? So to address this research question, we use the health and retirement, the 2060 wave of HRS, and then we link into the life history mail surveys in which we provide a lot of information about those people's, their lifetime job history. And then we link this data with the VBS in which provide us with epigenetic clock. And then we apply a bunch of sequence and cluster analysis to actually identify whether there's a difference in employment trajectory. And then we perform a series of regression and interaction, actually evaluate whether there's a difference across race and ethnicity. And we did found that there's 4 different employment trajectory over the life course. For example, the first one, those are people who experience long-term and stable, full-time job over their life course. And and then second group of the people, they have late labor force entries. And the third group are those people who experience multiple job disruption over their lifetime. And then the final one is the group that they experience long-term non-employed. And then we try to use race and ethnicity to predict which group are more likely to fall into this trajectory. And we did find there's a racial and gender difference. And for example, white male, they are more likely to have full-time job over their life course. And Black American, they are more likely to experience job disruption and also long-term non-employed. Then our biological aging here is measured by 5 different epigenetic clock, which include the first generation and the second generation clock. And then we did found that Black American, they tend to experience age acceleration in terms of the pass of age clock and then the green age. But somehow they experience slower age acceleration in terms of the Hunan clock. So there's a difference when we use different measurements. However, we found that compared to those people who experience a full-time job, actually people who experience multiple job disruption, they tend to have slower age acceleration. That's was a bit surprise to us, but we did find that Black American, even though they still show epigenetic age slower compared to when they experience long-term non-employed or multiple job disruption. So that's the highlight that we found from the studies, and I will be happy to talk more about this in our section. Thank you.
Thank you, Zoe. Finally, we have Wesley Wang.
Okay. Okay, good morning, everyone. Today I'll be presenting on a paper which I'm currently working on with my very brilliant supervisor, Eva Akimova, seated behind, in which we are trying to investigate the role of epigenetic aging, and how it fits into the relationship between childhood adversity and a range of, different known risky outcomes. So what we know about the relationship between adverse child experiences so far is that, we know that they tend to lead to a broad range of different deleterious or negative outcomes. For instance, they suffer, poorer physical health, they suffer poorer mental health, and they also tend to progress through the life course at an accelerated rate, meaning that they get married earlier, maybe they have children earlier age at first birth, and a greater number of children ever born by a younger age. And in the literature so far, we also know that, adverse child experiences are also related to an accelerated pace of epigenetic aging. So usually, the more adverse child experiences that you have experienced, or the more severe your adverse child experience is, you tend to accelerate more quickly in terms of your epigenetic age relative to your chronological age. So in trying to build onto this, literature, we're trying to actually move on from the idea of focusing on just the cumulative risk of child adversity. We're not just looking at how many adverse child experiences leads to how many increased or accelerated pace of aging. We're trying to incorporate the idea of timing effects as well, going from a life course perspective. Could it be that certain people have different sensitive periods, and that is what really matters between the relationship between adverse child experiences and epigenetic aging? Or could it be actually the recency of exposure that actually matters the most, rather than the cumulative risk or the cumulative counts of adverse child experiences? So that is what we're trying to explore in our very first research question. The second research question is basically trying to identify, if accelerated epigenetic aging do predict, a huge range of, health and mental health outcomes, as well as life history outcomes. And by life history outcomes, I mean what I've said earlier about progressing through the life course at an accelerated pace. And the third question is sort of like a blend between the questions 1 and 2, in which we investigate accelerated epigenetic aging as sort of a mediator. How does it fit into the relationship between adverse child experiences and the outcomes that I've mentioned in my second research question? Oh, and I should mention that the data set that we are using for this is the Future of Fragile Families and Child Wellbeing Study. You could see that my independent variable are taken from wave 3 to 5. And the mediator variable, which is the epigenetic clock, is taken 1 wave later in wave 6. And the outcome variables, which are the various range of health, mental health, and life history outcomes, are taken in the final wave. So this is temporally staggered, as you can see, to prevent reverse causality. So I'm just going to hop over to some findings very quickly. In the first research question, we did find that more than cumulative risks, which are usually covered in the literature, we do actually find that sensitive periods matters quite a bit more than cumulative risk. But to find out which sensitive periods, I'll be very happy to have you at the poster afterwards. And in the second question, I think this is quite in line with the literature, so I don't think that this should be surprising to find that actually accelerated epigenetic aging do positively predict a range of risky outcomes, and again, but not all the outcomes. So again, to see which outcomes really, are a function of accelerated epigenetic aging, please do visit the poster afterwards. And in the last research question, we do find a mediation effect for accelerated epigenetic aging for outcomes that we realize are actually more physical in nature rather than those that are more emotional and social in nature. And yeah, that's really all of my presentation. So thank you very much.
I could have my next 4 flash presenters come up, and if I could have Raphael to the podium, please.
Oh, nice. Hello, everyone. My name is Raphael. I'm a postdoc at Purdue University, and today we'll talk about my work for a while. So let's start with the research question. So we come up with this research question. It is, can we collect information during adolescence to make predictions in adulthood on a person? And basically, we can split our program in 3 different steps. The first one is collect information during adolescence. And for this case, we use AdHealth because AdHealth is a longitudinal program cohort. That means they have multiple waves, multiple collections of waves and information. And then we can have a machine learning model to try to predict and come with the best results. In our case, we did assemble. We did 5 different versions of machine learning. And then the output is basically we can have the prediction. We can predict the perceived symptoms of someone in adolescence and also in adulthood. So let's talk about more into details on exactly what I mean. So in our input, we have 3 different inputs from AdHealth data. Everything is collected during adolescence. So we can have only environmental data related to the perceived symptoms. We can also have only polygenic scores related to the perceived symptoms, or we can collect both of them having a polygenetic and environmental effects. And also, we want to see how sex is affected. So we have a stratification between only males, only females, and also both of them. So on our case, we have 9 different inputs in our model. And for that, we did 5 different machine learning models. We did a logistic regression. This is the most basic machine learning model you can do. And also, we go up into the scale of complexity into the deep learning of neural network. So I'm not going to show you what the best results are today. So if you want to see how I did and how is the best and the worst results, please come out on my poster, and then we can talk about it. And also for the outputs, we have 3 different outputs. We can compare for the perceived symptoms in the first wave when they are adolescents. They fill out the form with CDs19, the perceived symptoms question. So we made a sum up to see if they belong to the higher, the perceived symptoms category, or the lower symptoms, so we can make a prediction if they have or not the highest the perceived symptoms. And also, we can redo this for the wave 4. It is roughly 20 years later, and wave when they are roughly like between 40 and 30 years old. And also, they repeated the same CDs, but in a reduced, only 10 questions. So, and also during the wave 4, they also ask the clinical diagnosis. And also have the clinical diagnosis, and then we can compare the depressive symptoms, and also we can compare with the clinical medical diagnosis. So in between the 9 different inputs, the 5 different machine learning methods, and the 3 different outputs, we have more than 100 combinations in between. So again, I'm not going to show you, but if you guys are interested, we can talk more later. But I want to show you one quick results that we had. I think this is super nice. So basically, this graph means that the features are the questions in the questionnaire during adolescence, and the higher the bar, the most important are. So in blue is for when they are adolescents, and in red is when they are adults, 20 years later. So as you can see, our model it has an agreement between adolescence and also adulthood. They have like a fewer difference in between. You can see the top 3 are the same. And also, it's interesting to notice that in adolescence, you have questions, for example, if you want to leave home. This is like such a typical adolescent behavior. And also, you have another one that you have doubts, self-doubts, like, does everything just right? So you have like typical adolescence questions and feelings. And when you're an adult, this shift a little. So you have more concerns about physical health, for example, the frequency that you have urination, and also feelings about safety in your neighborhood. So it's interesting to have these different outputs if you are like a child with adolescence or if you are adult. So again, if you want to see more results and also if you want to see the polygenics impact on this predictor model, please come to see my poster. So thank you. And thank you everyone that worked with me.
Thank you. Next we'll have Moise.
All right, thank you so much. My name is Moise Bai. I'm an associate professor at the University of Arkansas at Little Rock and the University of Arkansas for Medical Sciences, so I'm in economics and health policy. This is one of my papers from grad school that I've just put away and I've come back to. So I'll be talking about genetic signatures of non-random selection into the military draft. So I'll be exploring social policy using the Wisconsin longitudinal study. So in the United States, from 1939 to 1973, the government had a draft. Any shortfalls in the personnel power for the armed forces were made up from having a draft. And so the draft was done locally by draft boards. So there was a big concern, or at least it was publicly understood, that the draft was not randomly choosing people to serve in the armed forces. So the nice thing about using the Wisconsin longitudinal study is it's a very long cohort study with a cohort born in 1939. And it has a rich set of background variables from when these men were in high school and so forth. So I have a rich set of early variables, imperfect variables such as IQ, and then measures of family background. The nice thing about the Wisconsin longitudinal study is that it also has collected recently polygenic scores. And so these are polygenic scores on other measures of ability, such as three personality traits from the 5-factor model of personality. So I have these early measures and I have these late measures, and I want to explore how people who were drafted or how men were drafted are different than men who voluntarily joined the military versus the men who never joined. All right, so that's my first question. Right now it's just about looking at non-random selection. So I have these multiple measures, and I'm using the Wisconsin longitudinal study. So it's a cohort study. It's not representative of non-white individuals. So it has a measure of IQ. It has some background measures. So the big issue with the more recent measures, such as polygenic scores, is that because they were measured late in life, obviously there's going to be attrition bias. And what we know about the evolution of genetics is that, we just can't, we don't want to think of this as a clean measure. So I acknowledge that this is, confounded by lived experiences. So if you were drafted, you might have had a completely different trajectory. So this is an imperfect measure, but it still can potentially help triangulate with the early life measures and the late life measures of potential non-random selection. All right, so unsurprisingly, based on IQ, men who are drafted statistically had a statistically significantly lower IQ by like 2%, not 2 points. They came from lower SES family backgrounds. So this is unsurprising. And then, so I have two of the three measures. The people in the military are statistically different on polygenic scores on a couple of the 5-factor models of personality traits. So the issue is the men, when we go beyond non-random selection, when we actually anchor into a real-life outcome like earnings, so even though men who are drafted are statistically similar to men who voluntarily joined, men who voluntarily joined have an earnings premium. Men who were drafted have an earnings penalty. So the next phase is to look at what causes, or try to explain some of these differences. And the idea is to look at maybe if polygenic scores can explain why people that are statistically similar on many observable characteristics yet have very statistically different outcomes on a meaningful measure such as earnings. So this is exploratory work, this is descriptive work. So far I show that draftees are statistically different than people who weren't drafted. So I'll stop here. Thank you.
Kyle, thank you.
Hi, everyone. So we've heard some talks where people have used epigenetic aging as an outcome, and actually here I'm going to be using epigenetic aging as a predictor, specifically looking at the onset of chronic disease, morbidity, and mortality among U.S. veterans. So just some very quick background. In this, I was interested in looking at whether epigenetic aging, as measured by Dunedin pace, would be predictive prospectively of the onset of chronic disease, measured using the electronic health record from the VA. The sample here was just under 2,200 US veterans, an average age of around 37 at baseline, and we had 13 years of electronic health record follow-up on average. So we were able to actually see of individuals who came into the study, they were enrolled between 2005 and 2016, what actually happens in terms of their chronic disease over time when we take the measures of epigenetic aging and predict forward. Hopefully what we're gonna see is that there's actually the onset of chronic disease that we expect. I'll note here that this kind of validation work was done when the epigenetic clocks were originally designed, but what's really cool to me about this is real doctors in a real medical setting giving real diagnoses to veterans. So this is a real world sort of application of where we might see these epigenetic clocks used in the future. So the major outcomes here, I'm gonna mostly focus on the Charleston Comorbidity Index, but and my poster will have all these other outcomes as well, including specific disease outcomes. You can see the diseases over there, really all the chronic diseases we care about. So what were the results? What you're seeing here on the y-axis is the Charleston Comorbidity Index. You can kind of think of this as you're getting 1 point per major chronic disease you get. And things like metastatic cancer, you get more points. But in general, this is a 10-year predictive mortality score. And higher scores, more chronic disease. So what I did was I just, for visualization, created a quartile split. So these are 25% groups for different Dunedin pace of aging. And what we see at baseline is exactly what you might expect. There are differences in chronic disease among the veterans by their Dunedin pace at baseline. But what's really interesting is what happens over time. We actually see trajectories of chronic disease onset that are greater among those who at baseline had faster Dunedin pace aging scores. So I'm putting up on the right here, this is a nonlinear scale. When you look at the Charleston Comorbidity Index, this is the predicted 10-year mortality risk associated with these scores. I have the actual mortality results at my poster, but I wanted to just pop real quick to diabetes. So diabetes was one of the diseases that had the highest degree of onset in this population. Again, age 37 to 50 on average is what we're looking at here. There were about 600, 700 cases in this cohort. I can give you the exact numbers afterwards. But what I'm showing here is a survival plot. So we have no veterans with diabetes at baseline are included in this plot. And again, I'm gonna show you these 4 quartile split. Over time, we're seeing that onset of diabetes is greater among the veterans who had faster Dunedin pace aging scores at baseline. Those are the hazard ratios if you want to actually see the differences in groups. But just absolute differences, right? Like, let's look at what these percentages actually look like. There's an almost 3.4 times absolute difference of onset. If you go to the right side, right, it's almost 30%, just over 30% in the fastest aging group, and it's about 9% in the slowest aging group. I'm working on some work right now showing that Dunedin pace is actually predicting the trajectory of A1C in this population as well, using clinical markers assessed in the EHR. And so if you also want to see results for PC Grim age, I have those at the poster as well. But I think the bottom line, what we're seeing is that the kinds of epigenetic scores that we're using now are actually predicting the things we want them to predict in real world medical settings, which makes it possibly good evidence for using this as a clinical surrogate health outcome in things like randomized clinical trials or other kinds of clinical settings. Thank you.
We have Ryan Frazier.
Oh, you control that. Hello. So I'll skip the title just to save 5 minutes, but I'm joined on this work with Jonathan Beauchamp, Jennifer Bernard, and Amanda Kowalski, and we're economists all, so I look forward to hearing the feedback this group has. But the core of this project is that we're going to use the universe of American mortality records to estimate the sibling resemblance and heritability of early life mortality in a modern American cohort. We see this as complementary to 2 main literatures. There's a lot of work doing sibling resemblance and heritability on completed longevity, but mechanically this is restricted to older cohorts, and it's going to find surprisingly low levels of similarity in completed longevity. Black et al. Is about 0.09 for reference. And one reason to think that this might be different in the modern context is advancements in healthcare have potentially increased the returns to socioeconomic status and health. Additionally, there's been some early work using similar methods to ours by Lacani et al. To estimate heritabilities of early life health conditions and medical claims data. But happily, mortality is a very rare outcome, so there are very few data sets that are able to actually have the power to estimate it. The data that we're going to bring to bear is the universe of American households in the 2000 census and their family relationship information, and we're going to link that to the universe of their Social Security Administration death records. So before getting into heritability, the main stylized fact that I'm going to present today that I think is new to the world is the sibling resemblance in early adulthood mortality by different types of siblings. And so what so this graph is going to show the sibling resemblance in age-specific mortality, defined as death on or before a given age. And it's going to be from ages 20 to 35 years old. And it's going to be separated by different types of siblings, namely same-sex and opposite-sex twin and non-twin siblings. Whereas the graphs are just split up to make it look cleaner. The female-female same-sex pairs are on the left graph, and the male-male same-sex pairs are in the right graph. And there are two main things to take away from this table. First, same-sex twins are just by far, even with these wide confidence bands, the most similar with the correlation on the binary outcome. And this is important, as you probably see where this is going, in estimating heritability, because same-sex twins are the only sibling type here that have identical twins among them. The second thing to note is that the resemblance is just shockingly low across all other types of siblings. This is like really, with sort of the exception of like maybe the male-male siblings, like male-male twins are going to be the most similar. They're going to peak out at about 0.01, just resemblance in the binary outcome. But everyone else is going to be less than like a 0.02 correlation in this, even throughout like the whole data period, which is going to be sort of suggestive of low importance of household factors. So to give a sense of how we're going to just estimate heritability, it's very similar to the framework, it's the same framework I presented at IGSS for those who attended, but we're basically just going to make a simple addition to the ACE model in which we allow twins to share more environmental similarities per specific factors B. And this is going to give useful identities of the resemblance in mortality row by different types of siblings, with the four main categories being same-sex and opposite-sex twin and non-twins. And I'm just going to collapse this to only look at female-female twins because the case of male siblings is analogous. Just to give a sense of what we're doing here, in the right column, we're just going to allow male-female twins to not share the same genetic importance of heritability and environmental factors. And we're going to allow for the correlations between these factors that are important to be imperfect, which is what the R's terms are. They're weakly less than 1. From top to bottom, we're going to allow twins to share more similar environments than non-twins, which is going to be what the importance of B squared term is, which is potentially important in the earlier life mortality that we'll look at in the future, which may be affected by uterine environment that twins share. And lastly, we allow same-sex twins to be the most similar sibling type, which is going to be empirically true, as we just saw. And they're going to be the most similar due to the extra genetic similarity this group on average has, which is weighted by the fraction of monozygotic twins that are among them, which is P. And in this framework, heritability and all the other parameters of interest can just reduce to this relatively simple expression, which is just the difference in the difference of the resemblances between sibling types. Happy to talk more about the estimation and what else we plan to do with these data in terms of heterogeneity going forward. Thank you.
Our second Flash Talk session soon, as I see people strolling back in from lunch. So first off, can I get our first 4 presenters up on the stage? We're going to follow the same format as right before we braked with the first Flash session. So Rosella, Giovanna, Nicolau, and Megan. All right. Well, I hope everyone had a very nice lunch before hopefully any inclement weather hits. Hopefully it won't hit. My name is James Lee. I'm a professor in the Department of Psychology here at UW-Madison. And I'm really excited to oversee our second Flash Talk session of this wonderful conference. This is going to be about genes and environments, G by E, and all things related to that. We will have 8 Flash speakers for this session, and I have, 45 minutes. So each speaker will be up here for 5 minutes to give an overview of their talks. Just as before, we'll hold all questions for the poster session that will immediately follow at 1:45 in the atrium that we were just at. Okay, so I guess without any further ado, I'll have the first speaker come up. The first speaker is Rosella de Sabata. Come on up.
Hello, thanks to TAGC for having me. In the next 5 minutes, I'm going to introduce you to my first PhD project, which also marks the first time I use genetics in my research. So the broad question I addressed is, what are the long-term health and cognitive effects of intrauterine exposure to low-dose radiation? The method I used to approach this question comes from my background in economics. So I leveraged the a plausibly exogenous spatial temporal variation induced by a smaller nuclear accident that happened in the UK in 1957, which is known as the Windscale fire, because a fire burned for around 20 hours, releasing different radioactive particles in the atmospheres, including, for instance, radioactive iodine. I used 2 main data sources to explore this question. One is the UK Biobank for my health and cognitive outcomes, as well as for genetics and other covariates. And the other one is the radioactive dispersion of the particles modeled by the Cambridge environmental consultants. And I linked the two at the individual data with the place and date of birth of the individuals in the UK Biobank. So my main effects are maybe somewhat not too surprising. I observe an increase in classically radiation-related outcomes. So for neoplasms, I observe a 5 to 15% increase for individuals exposed to the event in utero in the highest decile of deposition. And the range basically depends on the estimation method I use. I find that the effects are more marked for females, which is not in line with the developmental origins theory, which states that males are supposed to be more frail, but it's very much in line with the radiation literature, which has shown repeatedly that females are more radiosensitive. So in all of this, why genetics? So when I was designing my project, I really wanted to run a G/E study, also motivated by the research agenda of radiation protection research in Europe. Basically, the question is, how does sensitivity to radiation depend on genetics? What is the population heterogeneity? So based on this, and in absence of a GWAS on radiosensitivity, which doesn't exist to the best of my knowledge, I constructed, using the IU GWAS pipeline, some outcome-specific PGIs for my 3 main outcomes. Once I got the actual data, and I saw that my main effect is concentrated only in the highest decile of exposure, I really started to doubt my statistical power. So I report that I find a marginally significant negative G/E for neoplasm and cancers, and instead a positive one for thyroid neoplasms, but I discourage overinterpreting them. Thank you. So after this failure, let's say, so I was thinking of how to incorporate genetics to enrich my project, but I concluding that the G/E was not a good way to do so. I figured that genetics could help me explore from another angle an identification concern of my main estimate. So when studying events that have negative consequences for people exposed to in utero especially, it's a concern that the effects might be underestimated because the most frail individuals might have died before observation. So to address this, I use my outcome-specific PGIs, but as well as PGIs from the SSGAC to investigate whether they are predicted by my treatment status or whether, when included in my May regression, they affect the magnitude of my main estimates. And I do not find evidence for this in any of the two methods. So a third way that I've been thinking recently, whether genetics can tell me something, I'm always looking for alternative explanations of of my results, especially possibly correlated events that might drive the observed results. And although to the best of my knowledge, there's none, I would like to further investigate a theory from epidemiology, which is called the population mixes hypothesis, which suggests that increases in cancers might be related to migration. And I think that using genetic ancestry data might partially help me address this question. So please, if you have any knowledge about this, come talk to me during the poster session. Thank you.
Yeah, thank you very much for having me here. My name is Nico. I'm a postdoctoral research fellow at the University of Bologna. Did join work with Pietro Biroli and Elisabetta De Cao, also of the University of Bologna, Massimo Nelli of Turin, and Massimo Nelli of Bocconi and Silvia Mendoli of Turin. And the title is Tracing the Genetic Footprints of the UK National Health Service. And basically what we're doing in this paper will be showing that the national health system, the NHS in the UK, dramatically reduced mortality, and that this had an impact that can be kind of captured in the genetic predispositions of this population. So basically, it literally kind of changed the genetic structures of the UK population. So for the ones who are not familiar with the introduction of the NHS, the National Health System was one of the first kind of universal health coverage policies ever implemented in July 1948. So basically, from one day to another, like individuals in the UK became eligible to healthcare services free at the point of use. And what we will be kind of leveraging information from will be on pregnant mothers who from one day to another became eligible to deliver at a hospital setting. We all know pregnancy is a very crucial moment of life. Many things can go wrong. And we will argue that this reduced infant mortality rates by a lot, and that this actually led to some genetic differences that are discernible before and after the implementation of this policy. So in terms of the main results, as I was saying, we will see some clear kind of short-term effects, kind of huge effects on infant mortality rates, kind of 6.5 less deaths per 1,000 births, so a decline of 18% compared to the mean, which is massive. And this kind of leads us to the kind of next point. We say like these individuals who now survived are likely to be the most vulnerable ones who were dying before in absence to kind of being delivered at the hospital system. So they must have been genetically very different. And therefore, we will explore if this actually left a footprint in this kind of genetic structures. In exploring kind of a group of PGIs, we will see that after the implementation of the NHS, like there's a kind of consistent trend across PGIs with changes around 5% of a standard deviation. And finally, this is very exploratory analysis. We will be exploring if when we look at kind of long-term effects, this genetic information can be used to potentially attenuate, correct, for potential survival bias that could be introduced by this selective survival. So in terms of results, I will just show you kind of two figures. First figure, I want to just show you that basically the NHS had big effects on infant mortality rates. So first of all, you can see these are infant mortality per 1,000 births over time. And what you can see in these kind of dotted lines are the several events that occurred during World War II that affected infant mortality, but I'm interested actually in this red line, the implementation of the NHS, and you can see that within a 2-year bandwidth of this policy, infant mortality rates reduced by 6.5 deaths, 18% compared to the mean, if I run like an RDD on their local randomization. So I was saying like very huge effects. So what we do at the next step, we use, we run these models. Basically, we use UKB data. We use a group of different PGIs as outcomes. So basically, what you're seeing in this table is a forest plot with 19 regressions with these 19 PGIs as outcomes, and we explore if actually the prevalence of these PGIs differs pre-post NHS. And what we will be observing is a very clear trend, which let's say more qualitatively detrimental PGIs having higher values after the introduction of the NHS, while let's say more positive beneficial outcomes having lower values after the introduction of the NHS. And by positive or negative, I'm talking about depression, COPD, ADHD, and by kind of beneficial kind of phenotypes, I'm talking about subjective well-being, life satisfaction with finances, self-rated health. Effect sizes, as I was saying, they range between -5 to 5% of a standard deviation. We repeat the exercise with a subsample of siblings of the UKB. And basically, in this model, we can include this kind of family fixed effects. We have much bigger estimates, also much more noisy because the sample sizes are much smaller, but we obtain very consistent results. So if you're interested in this project, like please come to my poster and like I can walk you through the rest of the results. Thank you very much.
Not sure what, okay, there we go. All right, hi everyone. My name is Megan Cook, and I am a research associate in the Genes, Environments, and Neurodevelopment in Addiction lab, led by Jessica Salvatore at Rutgers University. And today I'm going to give you a very brief overview on a project I'm leading that's looking at the interplay between partner substance use and genetics on an individual's alcohol use. So we know that alcohol use is influenced by a combination of biological factors, such as genetics, and also psychosocial or environmental factors. For the purpose of this project, we're really focused on romantic partners and romantic partners' characteristics as that kind of psychosocial or environmental impact. And why is that interesting or potentially important? So we know that about 2/3 of Americans are either married or cohabit with a partner. And among those that are married, they spend 80% of their leisure time together. So this is an important person in these individuals' lives. So our main research question here is, if and how partner substance use moderates the association between genetic predispositions and alcohol use. To answer this question, we use data from the Collaborative Study of the Genetics of Alcoholism, or COGA. This is a multi-site, multi-generational study that was developed from probans who were in treatment for alcohol use disorder. And again, we limited the sample, we're interested in partners, we limited the sample to all individuals who were either married, engaged, or dating exclusively. And participants reported on their own and their partner's frequency of alcohol use, drunkenness, heavy episodic drinking, and cannabis use. For the genetic influences, we used summary statistics from a GWAS of drinks per week to create a polygenic score of alcohol consumption. And we used mixed effect models to account for the known familial nesting in the COGA data set. And included covariates of sex, age or birth cohort, marital status, so just a binary of whether or not the participant is married to that partner that we're interested in, and length of relationship as covariates. All right, so overall, looking at gene-environment correlation, so that is to what extent is the participant's polygenic score associated with their partner's substance use, we actually found no evidence for genetic correlation. But we did find that partner substance use moderates the association between the participants' genetic predisposition and that association with their own substance use. More specifically, we found that when the partner drinks more frequently or engages in more frequent heavy episodic drinking, the relationship between the genetic predisposition and the individual's own frequency of alcohol use and frequency of drunkenness is strengthened. And then conversely, when the partner uses more cannabis or uses cannabis more frequently, that association is weakened. So overall, I'm kind of here to say that just like we focus on parents in childhood and peers in adolescence and young adulthood, romantic partners are really a key potential influence to study in substance use behavior and trajectories of problem alcohol use in adulthood. And with that, I just want to thank the GINA team and the COGA participants.
All right, let's give another round of applause to our first 4 Flash speakers. Very nice. And I'd like to invite the next 4 up here on the panel. Erin Lumpy, Evelina Akimova, Li Chendong, and Yucheng Wu.
Oh, okay. Just go to the next one. Hit it. Hi, everybody. My name is Erin Lumpy. I am a PhD candidate at Rutgers. I work with Megan in the GINA lab. Thank you for allowing me to share my work today. I'll be discussing the legacy of parental alcohol use disorder. Parental alcohol use disorder, or AUD, is common, and approximately 20% of individuals in the US grow up with an AUD-affected parent. Parental alcohol use is associated with a variety of negative outcomes, including increased substance use across development. However, not all offspring with an AUD-affected parent will develop substance use problems. Let's take, for example, 2 popular celebrities. Both Jamie Lee Curtis and Sarah Jessica Parker have a parent with an alcohol use disorder. Both celebrities are female, approximately the same age, and work in the same profession. However, Jamie Lee Curtis has struggled with substance use problems in adulthood, while Sarah Jessica Parker has not. So why are some individuals with an AUD-affected parent, such as Jamie Lee Curtis, more susceptible to increased substance use in adulthood than others? One possibility is that the rearing environment associated with parental AUD may moderate genetic liability for substance use through gene-environment interaction. We tested this hypothesis by examining potential interactions between parental AUD and polygenic predispositions to alcohol misuse, cannabis use, and nicotine use in adulthood. We used both a population-based sample of Finnish twins and a diverse US-based sample enriched for familial history of AUD to test for generalization across populations with varying levels of genetic risk for substance use problems. Given previous literature demonstrating that adverse family environments in childhood increase genetic influences on offspring outcomes across the lifespan, we expected significant gene-environment interactions would align with the diathesis stress model, which posits that associations between underlying liabilities and outcomes are more pronounced under stressful environments such as parental AUD. In line with these expectations, we found evidence that parental AUD increases the positive association between polygenic predispositions and adult substance use. This interaction is visually depicted in the figure to the right. We see a positive association between polygenic predispositions and adult substance use for offspring raised by parents with the lowest number of AUD symptoms. However, this association is more pronounced for individuals raised by parents with the highest number of AUD symptoms. Some, but not all of these gene-environment interactions were replicated across samples. To learn more, please stop by my poster titled, "Does Parental Alcohol Use Disorder Moderate Polygenic Predispositions to Predict Substance Use?" The pre-registered analytic plan and findings can be found on the Open Science Framework by scanning the QR code here, and it is also on my poster. Thank you again for your time, and I look forward to speaking with you all further.
Hi, everyone. Before I start, I just want to thank everyone who's been organizing it, and especially can I say special thanks to Mindy, because she's been so awesome and helpful. So I just, and yeah, it's amazing to see. Should I press something or? I'm going to talk about intergenerational transmission of fertility. Great, sorry guys, thank you. So yes, this is joint work with amazing Agnes, who just finished her PhD in Oslo, and also with Torkild and my previous academic family, Melinda and other people in Oxford. And basically, I'm not sure whether you know my research, but I've been really interested in intergenerational transmission of socioeconomic status, as more like trained as sociologist. But recently, we were more and more wondering, we do not know that much about intergenerational transmission of family size or of fertility, which is actually particularly interesting case, because I don't know whether you noticed, but even on population level, usually childhood family size you basically raised in quite well correlated with your own number of children, and some quasi-experimental designs even show that part of that correlation is causal. And then there is obviously a question why. And more traditional in demography and sociology, the main explanations were based on socialization theory and on cultural norms and shifts, especially, as you know, with fertility behavior. There were huge changes with during past 20th century. So basically, the factors that would explain that kind of persistent correlation between childhood family size and adulthood family size is inheritedly found within social environments and families. Then it was obviously also proposed that there might be biology at least play a bit of role, or might be not. And we have realized that this question was not answered, quite to our surprise. And I hope I a bit convinced you why it's important to study fertility and its intergenerational transmission. As you know, also, we found previously that there is quite kind of interesting genomic signals that would predict number of children you would ever have. So basically, we know on the other side that it's not only environment that shapes your preferences or abilities, how many children have, but biology does play a role. But it all overly complicated because of its interconnections with for fertility ones with educational attainment, because as we all know, if you have higher education, you're postponing having children, so number of children also might be different. So basically we were trying to unpack this complex puzzle, believing that maybe if we have also biological information, that can be useful. So here, instead of text, I thought I'll just draw you what we have done, because we're interested at first whether childhood family size observed, would it predict adulthood family size when you control for different polygenic scores for fertility? So basically, is there any confounding? We're also curious whether it's other way around, basically whether childhood family size would basically confounds the link between polygenic scores and adulthood family size. And because of high correlations between behavioral, especially externalizing behavior, and educational attainment, as I mentioned, we also took lots and lots of different polygenic scores. And we have lots of set of controls. Please come and ask me about everything. And since it's also important, you know, like more lifestyle and life course trajectories, we were also trying to disentangle any kind of nuanced story, especially with regards to going to the university or maybe when you had your first kid, because that's quite important for the trajectory in the future. And we did lots and lots of regressions, but then I'm happy to actually discuss the methods at the poster. But what I want to say, I really like the data we used here. I don't know whether you used it's MOBA. It's the data from Norway. And Norway, in terms of fertility, study fertility is quite interesting country because they have amazing childcare, really kind of, it's its own world. But for science, it's quite cool one for this research question. And as you see, we have lots and lots of people who were genotyped, and we were observing them. Basically, we were able to observe them 40 plus and 45 plus. So anyway, and if you're wondering, let's do the results. But I thought since it's kind of prompt, it was more, yeah, I just want you to come and you will see what is actually happening. So yeah, thank you.
You hear me all right? Okay, nice. So, hi everyone, my name is Lee Chen-dong. I am a clinical psychology PhD student working with Dr. James Lee. And today, I will be presenting to you my project on downstream effects of genetic risk for externalizing behaviors, adolescent social environments. So a little bit of background. Externalizing behaviors are a constellation of acting out behaviors that are characterized by impulsivity and behavior under control. And some examples of externalizing behaviors are, aggression, rule-breaking behaviors, antisocialness, and substance use. So we know that externalizing behaviors have quite robust genetic influences, both from heritability estimates from twin studies, but also from genome-wide association studies, which has, the most recent one has identified more than 500 loci that are associated with externalizing. What we also know is that the genetic risk for externalizing behaviors are associated with environment as well. And in one part, in twin studies, we've seen that they have shared genetic variances between externalizing behaviors and psychosocial environments. And on the other hand, we have always seen, well, not always, but often seen association between polygenic scores for externalizing behaviors and psychosocial environments. However, I would say that less effort has been dedicated to exploring the mechanism or explanation of why the genetics and the environments are correlated. And especially now, we know that there are two possible mechanisms of RGE at play. So we have the passive mechanisms, which are essentially you're born into a more risky environment. So one example is genetic nurture, where you have parents and parental genotype that are somehow associated with your rearing environment. Population stratification is also an example. On the other hand, we have the non-passive, or I would argue more causal mechanisms through which the genetic risk of the adolescent or the offspring has a direct causal effect on the environment that they are in. So in this case, twin and sibling analysis gives us this unique opportunity to look at the contribution of passive versus non-passive mechanisms to the correlation between genetic risk and the environment. So here's the methods of our study. We used, research question first. So we want to test if we can see gene-environment correlations in the population, but more importantly, we want to see if the same effect can be observed at the within family level between twins and siblings. So we used the data from National Longitudinal Study of Adolescent to Adult Health. We used the data from Wave 1 when the participants are in their adolescence with an average age of 16. We elicited two separate samples from AdHealth. One is a population sample of unrelated individuals, but also a sample of dizygotic twins and full siblings. We calculated a polygenic score of externalizing behaviors based on a continuous shrinkage method and based on the most recent summary statistics for externalizing behaviors from a GWAS. And we listed 3 social environments in adolescence which has been proven to be risk factors associated with externalizing behavior and its development. So they are parental relationship, delinquent peer affiliation, and school connectedness. And finally, we fit, for the population sample, a multiple regression model, and for the twin sibling sample, a multi-level model with random intercepts to examine the within and between family effects of polygenic scores. So going to our results, we see lots of things going on here. So in the plot, you'll see the estimates of the effect of polygenic score on the social environments at the population level, within family, and between family. And I'm just going to quickly summarize what we see here. So first of all, we see that gene-environment correlation for externalizing genetic risk are actually ubiquitous in the sense that at the population level, we see those correlations across all 4 variables we used. And for parental relationship, more specifically, we see that there is a robust within-family association with polygenic score for externalizing behavior, which basically says, the twin and the sibling with higher polygenic score are subject to less supportive and less close parental relationships. And we think that within family effect actually reflects a kind of non-passive and causal mechanism. On the other hand, for delinquent peer affiliation and school connectedness, we see that the effect attenuated going from the population and between family level to within family level, which then may reflect the fact that the gene-environment correlations with respect to peer and with respect to the school environment may be due to more of a passive mechanism, or in other words, they're born into those environments. And finally, our study has implications in terms of the gene-environment correlation investigation in general. So we see that different environments may be attributable to different RGE mechanisms. And our finding may also, with respect to the parental relationship piece, has implication for the prevention and intervention for externalizing behaviors, because we see that genetic risk may accumulate outside of the skin in the form of social environment or environmental risks. And that's everything I have.
Hello, everyone. My name is Yuchang Wu. I'm an assistant scientist at UW Medicine. So after lunch, I see someone is getting sleepy. So I will share with you a very fun project, which is the genetic basis of misreporting height in all of us. So the central limit theorem tells us that for polygenic traits like human height should roughly follow a normal distribution like this. Here I made a histogram of height using the European samples in the UK Biobank. Different colors represent males. Different colors represent males and females separately. So in UKB, the height was measured by a nurse using a measuring machine following a standard procedure. Therefore, the height was measured very consistently, objectively, and accurately. So therefore, the distribution almost looks like a nearly perfect bell curve. However, it is well known that there are abnormalities in self-reported height. So here is the same histogram of height, but self-reported height from the CDC survey, where they surveyed for more than 60,000 people. As you can see, the distribution does not look very normal. Particularly, we see a very mysterious spike at 6 feet for men. So indicating there are more samples, there are more men self-report being 6 feet tall than we expected. So our question is there a genetic basis for this misreporting behavior? So in order to do this, we need 3 pieces of information. We need to have self-reported height, objectively measured height, and genetic data. In all of us, a subset of samples provided self-reported height, and nearly all the samples have objectively measured height, either through physical measurement or electronic health record. And a substantial proportion of the samples have genetic data. So therefore, we first compare the self-reported height versus the measured height. And here's the histogram of the difference using all the samples, the females and the males. And we find a significant overreport of height in males. And then we run a GWAS on this difference and to see whether there are genetics could contribute to this tendency to misreport. And we find a positive heritability estimate with large standard errors because of the small sample size. And in order to understand the genetic architecture of this misreporting behavior, we calculate the genetic correlations with 45 complex traits. Here are some of the results. And the more details will be included in the poster. There are two takeaway messages here. The first, we find some evidence for misreporting height in all of us based on the phenotypic level and also genetic level. The second take-home message is, if you ever work on the all of us data, you know that the data is really messy, even for a very simple trait like human height. And there are nuances in the data, and so therefore, whenever you work on the all of us data, you really need to be careful about the data. So that's all. Thank you.
All right. Well, let's give another round of applause to all of our Flash speakers for this session. And we will now conclude this session and go into the atrium for our poster discussion and break. Come back at 2:30 where we will start our next paper session.
Welcome back. I hope you have enjoyed the very nice discussions happening there. And I'm pretty sure we have plenty of time this evening and tomorrow to continue the wonderful discussions. So again, my name is Tianyuan Lu. I'm an assistant professor here at UW-Madison. I will be chairing this next session titled Big Picture. So this session is really about stepping back and asking big problems. So we ask about where we come from, how we study ourselves, and what it means to be human in the age of genomics. So you will hear from three researchers working across different disciplines. So we have evolutionary biology, social theory, philosophy, and behavioral genetics. So they really tackle fundamental questions at the heart of social genomics. So questions questioning the interplay between genes and environment, explanation and meaning, science and society. So our excellent speakers include Lucas Kasten from University of Iowa, Bennett McIntosh from our UW-Madison, and Damien Morris from KCL. So I hope these talks will challenge our assumptions, provoke reflection, and invite us to think more deeply, not just about the data we work with, but also the frameworks that shape our interpretation of the data. So let's welcome the first speaker.
Thank you for the kind introduction. I'm really excited to be here and share something kind of out there. So my name's Lucas. I'm currently finishing out my PhD at the University of Iowa. I'm in the process of looking for a postdoc. Wink, wink. But today I'm going to be talking about language evolution, but more broadly applicable. I'm also be talking about a new polygenic score-based approach we've been working on that tells us when complex traits evolved. But before jumping into that, I want to talk a little bit about why language is important to study. So number one, language issues are incredibly common. So about 7% of children have a language disorder characterized by either struggling to communicate or produce language, or struggling to comprehend language or produce language. And as you can imagine, these language impairments are pretty strongly linked to our social and educational outcomes. If you struggle to communicate, you're gonna have a harder time making friends. You struggle to comprehend language, you're gonna have a harder time in school. This can be related to worse educational outcomes down the line. And so while these language issues present very early in life, they have lifelong implications. And so recently there have been some really great language-related GWASs. They're still pretty small, but even with these studies, there's still a lot of big questions about language that we still don't know about, especially about its biology. My hypothesis is, I can put on my tinfoil hat, but my hypothesis is the more we learn about the origins of language, that'll provide a lot of new insights into social and psychiatric traits and potentially provide new interventions or behavioral therapies for these traits. So everything I'm going to talk about today is part of a much larger preprint, so if you're interested, please check it out or come talk to me. But here's the outline for this talk. So there's two key claims I'm going to try to convince you of. So number one, in the first part, I'll try to convince you that we have this new method that we're calling evolutionary stratified polygenic scores that tells us when a new trait evolved. And in the second part of the talk, I'm going to try to convince you that with this method, we find that language capabilities are pretty old, and that Neanderthals may actually have more language capabilities than most of us in this room. Jumping into our study, so we started off with a pretty small discovery sample with a really unique local cohort called EpiSLI. We had about 350 kids, but what's incredible about this study is they have longitudinal, really deep language and cognitive phenotyping. So we're able to use this really rich data set to quantify 7 different domains of language. So those are those factors there. And so while each one of those probably deserves its own talk, I'm really just going to focus on 2 of them. So we're really interested in factor 1, this core language factor. So that's the main trait we're interested in. But as a control to figure out if any genetic associations we find are specific to language, I'm also going to be talking a little bit about nonverbal IQ. And so while our initial discovery sample is pretty small, we validated this in a couple other cohorts, including a study of over 30,000 people. So, all right, so once we had those language scores, the next thing we did was just a basic polygenic score analysis. So you guys know what a GWAS is, you know what a polygenic score is, so I'm not gonna rehash that. But there's one thing I want to point out. So the traditional polygenic score methodology is genome-wide. And so that's perfect or great if you just want to maximize prediction accuracy, but that doesn't really tell you anything about the underlying evolution, right? So I'll talk in a minute about how our approach will address that. But here's a big ugly heat map of all our results from this genome-wide based approach. And while there's a handful of really interesting things here, there's only one that I really want to talk about for this presentation. That's this very statistically significant association between our core language factor and the cognitive performance polygenic score, and a relatively weak association between the same polygenic score and nonverbal IQ. And this is interesting because this gives us something that we can try to dissect evolutionarily to see, why is this polygenic score a little bit more related to language. And so to do that, we had to come up with a new approach. So this our new method. Again, we're calling evolutionary stratified polygenic scores. So it's a mouthful. If you have any recommendations, let me know. We'll probably go in the acknowledgements. But the basic idea is really similar to partition heritability, where we take our GWAS summary stats. These are the exact same summary stats you'd use for a polygenic score, but we add in a layer of evolutionary annotations. And so here's kind of a toy example here. But what's important is that now we can kind of partition the genome. So we can take everything not in the regions of interest that we're calling the background to compute a stratified polygenic score. So we compute the background polygenic score that we're going to control for, and then we can compute an evolutionary stratified polygenic score. So this is the part of the genome that we're really interested in that has been associated with some sort of evolutionary event, and you can use any evolutionary annotation you want. But once we do that, we will then statistically compare those two to see by modeling them to see if adding in this ESPGS term significantly improves prediction accuracy. And what this will do is figure out the evolutionary partitions of the genome that are most associated with a phenotype, and from that, that'll tell us, should tell us when a complex trait evolved. So this is a pretty simple concept, but we had to validate it, which is a big ask. So to validate it, we had to come up with a reasonable hypothesis that could be falsifiable. And so our hypothesis for language evolution was that genes that differentiate humans from chimps will be associated with language. So we compute this ESPGS in our EpiSLI sample. So the x-axis is that polygenic score, only using human chimp divergent SNPs. And y-axis is our core language score, so that factor 1. After we adjust for the rest of the genome, we do see there's a significant, relatively weak association, but that kind of confirms our hypothesis and tells us that our method actually works. So now that we have this method that works, we can expand to a bunch of different evolutionary annotations to really pinpoint when language capabilities evolved. And so there's a lot going on in this figure. It's really only one important thing, though. So we looked across 65 million years of primate and human evolutionary events. We found only one single annotation that was reproducibly associated with language. That was this thing called hackers. What was incredible is that this hacker polygenic score was very specifically predictive of language and had actually a correlation is 0 with nonverbal IQ, which is pretty remarkable. And at this point, you're probably wondering, what the heck are hackers? So this stands for Human Ancestor Quickly Evolved Regions. So these are actually the fastest evolving regions of the human genome. And what's really cool about these is they evolved before the human Neanderthal split, making them at least 500,000 years old, probably even older. But these are primarily non-coding elements of the genome, so they're mostly regulatory. They give humans new gene regulation capabilities. And so this is a relatively new annotation, just published a couple years ago in Cell. So we still don't know a lot about it. So we're the first ones to really strongly link this to language. But all those results were based off that ESPGS method. So we wanted to, again, try to validate that and prove that this method works. To do that, we thought that, well, if these regions of the genome hackers really did evolve to support language, then our ancestral reversion, so a mutation kind of back to the human chimp ancestor allele or like a gorilla ancestor allele should negatively influence language, right? We actually computed that. So looking at rare single nucleotide polymorphisms. So in everyone's genome, we can count these up. And what was incredible is that 99.99% of people have at least one of these reversions. So these are kind of hotspots for these ancestral reversions, and I'll talk about why we see that in a few minutes. But essentially, we can count these up in the hackers and other evolutionary significant regions of the genome, like HARS, and in just random sequence. That we can take those scores and use them to predict phenotypes. And so there's a lot going on here. But the key takeaway from this figure is that the more ancestral reversions you have, the later you develop language. So for every 3 reversions somebody has, the age they start speaking is delayed by 1 month. And so if we look at this distribution in D, we see there are a few people with 0, there's some people with 21. So if we look at those 2 extremes, we'd predict there to be about a 7-month difference on when they start speaking, which is a pretty substantial difference. And again, what's incredible is that this is specific to language. So it was not associated at all with age these children started walking or with broader intellectual disability. So again, suggesting hackers have some sort of specificity on language and not just general cognition. And so hopefully I convince you that we have this cool method and that hackers are really related to language. But now we wanted to figure out how these traits change over time. And since these regions of the genome evolved before the human Neanderthal split, they give us a really kind of crazy idea to try to predict what language and general cognition will look like in Neanderthals. And so we again compute this ESPGS in modern Europeans and in the ancient 4 high-quality ancient Neanderthal genomes. If we look at the background score, which would just be general cognition, we find that Neanderthals score really low in the 7th percentile. But what's remarkable is that Neanderthals actually score on average in the 82nd percentile for this hacker-based language-related polygenic score, which would suggest that you take a Neanderthal from 100,000 years ago, they may be able to learn language better than we do. And that's really unexpected and really surprising. We were pretty suspicious of this finding for a while. So the next thing we did is try to build on this and figure out what's going on. Was just ask the question, well, how is this, has language capabilities changed across more recent human history? So to answer that question, we again computed ESPGS, but this time we looked in ancient human genomes, ranging from the past 20,000 years ago to 100 years ago. So we had over 3,000 people here. What we find is that the background polygenic score, the general cognitive ability score, has increased a ton over time, meaning general cognition has been positively selected for, which makes sense. It's a good trait. It should be positively selected for. But what's shocking is that that language score, that purple line, is totally flat, meaning language capabilities have not changed at all, or genetically predicted language capabilities haven't really changed at all for at least 20,000 years. That leads to the obvious question of, well, why wouldn't it increase, especially if general cognition is going up so much? And so we looked around a couple different data sets, but we found one really intriguing possible explanation in the ABCD cohort. And so we essentially find this pleiotropy between these hackers and language, and also birth-related complications. So we did a within-family analysis here. What we find is that siblings that are born heavier carry more of this hacker polygenic score, which would suggest that more language capabilities is related to higher birth weight. And interestingly, we also found about 50 pairs of siblings, full siblings, that were discordant for C-sections, meaning one was born vaginally, the other was born by C-section. We find that the ones born by C-section have significantly more of this hacker polygenic score. So now when we take that into account, this figure starts to make a little bit more sense, right? So we kind of have these two opposing pressures. You have birth complications that are going to be selecting against this hacker polygenic score, but you have the fact that language capability is a good thing, so that's going to be selected for. So that leads to this pattern of balancing selection where they kind of cancel each other out, leading to this totally flat line that we see there. And so this suggests that language capabilities had a reproductive trade-off. And this last analysis is really out there, so I'll just go over it pretty quickly. But we were wondering, well, these sequences, at least similar regions of the genome, do exist in other species, so what are they doing? And so what we find is that there's evidence of convergent evolution. So essentially, we can find other mammalian species, about 50 of them, that have really complex vocal capabilities. We can compare them to other species that don't have complex vocal capabilities. What we find is that the animals like bats and whales and dolphins have convergently evolved similar hacker-like sequences, which would suggest that you take these similar sequences and you put them in any mammal, they might get better language capabilities. And again, confirming that these hacker sequences are somehow and for some reason important to language. And with that, I want to start to wrap up the talk. So there's 4 key takeaways here. So number 1, we have this cool new method. I'll let you guys be the judge, but we think that it's really exciting and it can identify when new traits or when complex traits evolved. Number 2, by applying this method, we found that language capabilities appear to be a lot older than a lot of people thought, and language capabilities predated the human-Neanderthal split, so they're at least 500,000 years old. It's important to note that doesn't mean that language is 500,000 years old, it just means the cognitive capability for that is very old. 3, we find that there's a reproductive trade-off with language capabilities, leading language ability to be stable for at least 20,000 years, possibly even older. And finally, we found evidence of convergent evolution across mammals, suggesting that these sequences are kind of like the universal substrate for complex communication. Okay, with that, I want to thank everyone who's a part of this project. I especially want to thank my PI, Dr. Michelson, who's been fantastic, and our funding source. And so, yeah, if you're interested in this method, want to apply it to another trait, please come talk to me. I'd love to support in any way that I can, and I'll take any questions.
Thank you very much. We have time for several questions.
Thank you for a really great talk, Lucas. That was fascinating. I'm curious if you could say a little bit more about these hacker sequences in terms of the biology of what you find in there. Are these long non-coding RNAs? And can you see, are they like expressed in the brain if you look in GTEx or other repositories?
Yeah, so that's a really important question. So we have a whole kind of subsection in our preprint about the molecular effects, but essentially what we find is that these are mostly non-coding regions. So they're mostly regulatory. They're enriched for single cell, or essentially just QTLs. So they are enriched for variants that influence gene expression, and they're really enriched for prenatal gene expression and not postnatal. So this suggests that these regions of the genome are acting prenatally to influence later language capabilities, which we're still trying to figure out what exactly is going on with that. But we also did some other analyses that we find these sequences actually evolved in humans to better bind well-known language genes like FOXP2. So we were able to essentially link our results with what's already known about language genetics. But I think this is a little bit more applicable to the population level because FOXP2 variants are pretty rare.
Thanks for a great talk. So maybe I didn't catch this, but I'm just interested in where the Neanderthal and Denisovan data sets are coming from.
Yes. So those are very rare, but there's essentially 3 high-quality Neanderthal genomes, and they're from, I think, almost like 70,000 years split. So it's very messy. They're from, I think, mostly Eastern Europe. And it's made by the Max Planck Institute, and then the Denisovan genome is similar, but it also has, I think it's like a quarter of its ancestry is Neanderthal. So we only have 4 genomes, so we're making some pretty big conclusions here, but there isn't a lot of high-quality Neanderthal DNA.
Super cool.
Thank you.
I did not expect to see that coming to the Social Science Genetics Conference. That's pretty badass. I just had a question about what you're calling this PGS.
Yep.
So when you were giving the talk, I thought what you meant was that this is just a polygenic score for the number of sort of novel alleles a person is carrying. Did you identify the loci and then reparameterize on a language phenotype?
No. So yeah, good question. So we take the we compute it like the traditional polygenetic score. So we take the GWAS for cognitive performance, and instead of using the whole genome, we only use the SNPs that are like evolutionarily meaningful. So we kind of subset the genome.
I guess what I'm wondering is, so you're only interpreting the, I think people can hear me. Oh, yeah, so the Zoom audience. You take the results of the GWAS, you drop out all the SNPs that aren't affected by this evolutionary process, but don't each of those SNPs have an ancestral allele?
Yes.
And so what if the ancestral allele is not the cognitively enhancing allele, like where the sort of evolutionary loading and the trait loading disagree?
Yeah, that's an important question. That's something what we're starting to think about. So that was kind of the reversion analysis I showed, I think addresses exactly that. But the polygenic score-based approach is still very simple. So we're not taking that into account yet. We're just saying that this partition, like this chunk of the genome that we know is different between humans and chimps, is especially important for this trait.
I see. And so you take the observed loadings on the cognition GWAS.
Yeah, we just take,
I see.
so it's essentially.
So that's why it generalizes to any trait you might want to do this thing to. It's just say focus on the evolutionary novel stuff. It would be great to have some kind of negative control here where you had a trait that you didn't think like we were that different in relative to our primate ancestors. So like maybe a hematological trait or something pretty vanilla.
But
the other thing that occurred to me, like the talk started out and you were like, well, look, we have a stronger correlation with cognition PGI with verbal versus nonverbal. And I was like, oh, this is not going good places. Like that's about the test they gave them to generate the GWAS. And you took it somewhere amazing. So thank you.
Thank you.
But I would say just like keep that in mind that the trait that the thing loads on is going to be a function of the trait that the thing was trained on. And if you've got more verbal content in those cognitive tests, you're going to get this verbal skewed score.
That's very helpful. Thank you.
Thank you again, and for the very nice discussions. So we will next welcome the next speaker.
Great, thanks everyone. Thanks to Mindy and everyone else here at ISG for organizing this. My name is a secure connection with the site cannot be verified. My name is Bennett McIntosh. I'm a PhD candidate in the History of Science program here at UW-Madison. In addition to the ISG, which many of you know is a great cluster for social genomics research, UW-Madison also has the oldest program in the history of science at a public university in the US. And so I'm really happy to be part of that and able to interface with so many great colleagues here in ISG. I'm writing a dissertation on the history of data sharing practices in social science genomics. And I want to talk about history that's maybe not so ancient as Lucas is today, but about the 10, 20-year-old history of this field, how it evolved, and some of the implications for that for data sharing practices today. So historians of science don't just study what was discovered and when, and what the technical innovations that led to those discoveries were. Many of us are very interested in the social structures of science, the kinds of collaboration that can occur, how that leads to collaboration, innovation, which new fields are coming into a space like this. So social science genetics only recently has a lot of participation from economics, political science, sociology. What I'm interested in is something that kind of brings together these two different ways of looking at the history, which is the data practices within social genetics, the ways that people share data, produce data, cooperate to maintain and interpret those data. I think this helps to bridge our understanding of how the field developed, technically speaking, and how the social structure and dimensions of the field have evolved. So today I want to introduce you or reintroduce you to a critique that many of you may have heard from my colleagues in the history or the philosophy or the sociology of science, this critique of geneticization, which is this kind of over-indexing on genetics in terms of understanding human phenomena that many critics of behavior genetics and social science genetics think is present. And then I want to introduce I commonly hear scientists say that, oh, any hype or misapplication around over-applying genetics is really just the result of miscommunication. Whereas some of the more critical scholars who are studying this field might say that it's actually, there's something fundamentally wrong with behavior genetics, that a reductionist science is inevitably going to lead to overly reductionist public understanding of science. I think it's not just a matter of miscommunication. So your research itself does have some causal role to play in how people understand the science. Maybe that's not so radical that your public understanding of your research is caused by the research itself. But I also think that the influence of on public understanding of social genomics is less direct than just over-reproductionist research leads to over-reductionist understanding of science. So I want to introduce this term that I think better describes what actually happens day to day in social science genetics, reproductionism. Social science genetics builds reproductionist models that try to reproduce social phenomena in silico usually. And then some of the components of those models that are most influential function as what we call boundary objects. And I'll get more into those and then discuss what the implications of this are for interpreting social science genetics more broadly. So historians, sociologists of science, especially those who are critical of behavior genetics, social genomic findings, often couch their critiques in terms of geneticization. So this is, Abby Lippman was an epidemiologist who was critical of kind of over-indexing on genetics in terms of understanding maternal health, prenatal health. Who coined this term geneticization, but these critiques are much older than the 1990s. You might recognize a little bit of Lewontin or Levins in here as well. But critiques of geneticization can often be characterized, or at least caricatured, as three different maybe failure modes. The first is genetic reductionism, which is said to involve this over-indexing on one gene causes one trait. So that's about the cause that we expect to see coming from genetics. The second is determinism, that a set of causes, whether that's one-to-one or a broader set of causes, are said to lead 100% or deterministically to a given trait. And the third is slightly different. It's about the role that genes play in our identity. So genetic essentialism says that my ethnicity or who my kin are, or who my people are, or my biosocial identity. So my identity as a previvor of cancer, or my identity as someone who suffers from whatever biological trait is tied directly to my genetics. I can guess that a lot of you in here are thinking that genetics currently is not actually that deterministic. There's lots of probabilistic work that recognizes a lot of environmental contingency, and very few people in here are going to say that who I am is defined entirely by my genes, so genetics isn't essentialist. I think that's largely true. I think this 35-year-old critique has been updated a little bit, and I'm happy to talk more about that. But you might also be thinking, well, wait a minute, reductionism, isn't that what we want to do in science? Don't we want to analyze causes, take them apart, find the individual components that are bringing things in? I think that's true. Genetic reductionism should be distinguished from reductionism writ large. Reductionism writ large obviously includes other potential causal influences. But I think that the overly reductionist public understanding of social science genetic results, which many of my colleagues have said is the result of the reductionist tendency in genetics itself, I think it's not just a result of reductionism. I think it's a result of how social science genetics works day to day, which is something that my colleagues call reproductionism. So reproductionism is a way that a lot of modern data-centric sciences work, where you're not interested in modeling the tiny components of every single cause and then bringing that together into a total model. But instead, let's take meteorology, for example. So in this great book on climate science, Paul Edwards talks about how meteorologists build their forecast models, both for the weather tonight when it's going to rain, or the weather 30, 40 years in the future through climate change. And to build those models to simulate what's going to happen, they bring together, yes, raw theory, but also data that they're collecting live and what Edwards calls semi-empirical parameters. And whenever you take you generate a PGI for one of your participants, or whenever you annotate someone as belonging to a certain reference set, I think what you're doing is saying, I have some knowledge about this person that is semi-empirical. I think their PGI, is one standard deviation above the mean. And that's not a purely theoretical element of your model. That's something that you're describing an individual based on your understanding of the population that you're coming from. And then you're using this mix of theory data and semi-empirical parameters to build your model of the population dynamics that are happening. What are the kinds of things that you introduce into your reproductionist models, into your directed acyclic graphs that we've seen up here earlier today? I think one of the most important kinds of model components for understanding the public understanding of social genetics is a kind of scientific object that we call boundary objects, because they exist on the boundaries of different social worlds, of different discourses. So the canonical example in this paper by Starr and Griesmer in 1989 is museum collections. The person who's selling an item to the museum, the person who's buying the item for the museum, the scientist who is studying the item at the museum, the administrator who's making sure that the museum has a valuable connection. All of those are going to have really different understandings of what the object is that is coming into the museum, but the object is stable enough, it coheres enough to make it recognizable between the different social worlds. And I claim that some of the most important tools, most important model elements for the public understanding of social genomics have this property of being boundary objects, of being these tools that allow collaboration across different social worlds without necessarily full consensus on what those are. And so the first of these are GWAS results. I'm borrowing some figures from some of the SSGAC's papers here. Many of you would not be here had consortium GWAS not led to positive results, to GWAS hits for social traits, right? We see as the sample size of GWAS, this one's for educational attainment, increases, so does the predictivity of polygenic scores. And then we also see on the right there, over time, the frequency with which behavior geneticists are using polygenic indices has increased dramatically over the last couple of decades. And this is because the polygenic index is useful in many different ways for many different things. People in this room may not agree exactly on how to causally interpret a polygenic index. They definitely don't agree on what they ought to be doing with a polygenic index, because there are so many different things that you can do with a polygenic index. Thank you. It can be an estimate of heritability or a covariate or a clinical tool. And because the different ways that you can interpret a polygenic index are so broad, I think it shouldn't actually be that surprising to us that more controversial, both in terms of scientific rigor and in terms of ethics, more controversial uses of polygenic indices are also fully within the realm of possibility, because there's so much interpretive flexibility around what it is that a polygenic index can do. I think the same is true for categorizing people into population categories on the basis of their genetics. Obviously, this is something that's very useful, that's kind of fundamental for imputation and understanding population structure. But at the same time, over the 2010s into the 2020s, a lot of researchers in genetics and a lot of institutions in genetics have kind of pushed back against certain interpretations of genetic ancestry, especially trying to separate our understanding of genetic ancestry from our understanding of race. Yes, these are related, but they refer to different concepts. And so, for instance, this Hastings Center report with several social and behavioral geneticists on it says that there's a risk of confusion between especially continental level genetic ancestries and racial and ethnic groups, and says that if you're going to download, say, SSGAC data, you shouldn't be doing certain things like making inter-ethnic group comparisons using these scores. I think for both polygenic scores and for genetic ancestry, one of the characteristics that make them most powerful are that you can attribute something to an individual, you can predict something about them with the score, or you can you can say something about an individual in any context. And I think that this has some really important implications for how the field is constructed. It's only because of the many different uses of modeling tools like polygenic indices and GWAS results and ancestry categories that we have some of the institutions that have given us the scale to do social science genetics, that also produces new possibilities for what it is that we can do with these scores that are not kind of well regulated, and the ways in which these ought to be regulated are still matters of controversy both within and without this field. This was kind of the historical development of social genetics, but that's only part of my dissertation. I'm writing much more about what's happening today, and I'd love to talk to you about your experience with people trying to maintain the context with which their scores or with which their data tools are developed, or also to work with tools that are maybe have some sort of data controls on them, for instance, in a research analysis platform or various terms of use. So that's my email up there. I would love to talk to you more. Thank you.
Thank you. And we have time for a couple of questions.
Thanks, Bennett, for that great talk. I wonder if you formed either weak or strong opinions of the use of FAQs in some of the science generated by the group here.
Yeah, I mean, I think FAQs are really interesting because they're like a non-binding way of providing more context for the tools that you're sharing with the world. So if you're saying, here is a method that you can use, but wait, don't use it in this way, or wait, maybe this is some of the things that you need to consider here. That does provide more freedom to do kind of new research, which binding terms of use or binding kind of research agreements don't necessarily allow. But it's also, people can just ignore FAQs. And that's especially the case for in kind of public discourse about these. Plenty of people writing maybe in the media about this won't even read the paper, let alone the 30, 40-page FAQ. So I think there's a lot of room to kind of look for new ways of making sure that things keep their context without being too restrictive. I think there's a question here.
Thanks. I appreciate the talk. I have to confess I'm always a little lost when I hear these humanities talks about genetics.
Very fair.
Right. And as I imagine many people from the humanities feel when they sit in a room and hear us talk about this stuff. So it was very helpful to hear some of your meta comments about, my field thinks this and I'm wondering about that. But I guess what I'm curious to hear your thought on is, what does your discipline want to get out of this engagement with this? It's not quite a discipline, but this body of work that it's concerning itself with. What is the? Yeah.
I mean, one of those is even figuring out what it means to be a discipline, right? Like history of science, most of my colleagues don't study social genomics or any contemporary science, but they're very interested in this question of how is it the disciplines form. And so you all are a case that we can use to kind of answer that question. But there's also, a body of work that is much more specifically critical of behavior genetics, hopes to add philosophical tools, to add critiques that maybe some of you will engage with. I think that work's valuable. I think especially the work that tries to understand kind of public interpretations of these results. To what extent is rhetoric in the FAQs? Is that ending up in online conversation? Is that ending up in news reports? Is that ending up in people's decisions about whether to use a PGI in embryonic selection? I think that's interesting and valuable. I'm trying to kind of start a little earlier in the process and understand where it is that any of these tools that someone might use when they're trying to understand genetics, where is it that they're coming from, and why is it that they're useful to you all? Because I think a lot of my colleagues say, well, this stuff can lead to bad results, therefore don't categorize people by ancestry at all. And I'm like, wait a minute, there's a reason that people are doing that here, and it's not just that we think that ancestry categories are kind of the end-all be-all of understanding genetics. There are kind of utilitarian reasons to do that. And I think understanding those dynamics and that historical development is useful for this sort of interdisciplinary rapprochement.
So just to clarify, the answer is that some of your colleagues want to change us, but you just want to know what we are.
I think I may come out of this wanting to change you. Jury's still out there. But I think I can also, and this is where these questions come in, I think I can also come out of this with advice about how to make sure that the context for these tools continues to travel with the tools while minimally restricting the science that is happening, because a lot of the ways that control these tools actually end up having consequences that make it harder to do science.
All right, thank you again for the interesting talk. And
I'm happy to talk during the poster session.
In the interest of time, we will have to move to the our final speaker for this session. But yeah, definitely go ahead and discuss after this.
Yeah, here's
Thank
some
you.
extra slides. It's right there.
Great. Good afternoon, everyone. I'm Damien Morris. I've just finished my PhD at King's College London. So this session is called Big Picture, and this talk, it doesn't get much bigger picture than this. I'm going to be spending my brief time with you today talking about free will, human agency, and personal responsibility, and discussing how these philosophical questions bear on how we do social and behavioral genetic research. This talk is based on an academic monograph that I wrote that's due to be published in Twin Research and Human Genetics. It's going to be spread across 3 issues. It's 50,000 words long, so I'm obviously only going to be able to tease you with a little part of it today, and hope it whets your appetite to read more when it comes out. But you might already be asking yourself, what has all of this got to do with me? I'm a scientist. I'm concerned with empirical questions, not these abstract philosophical questions. Well, it turns out it has quite a lot to do with us. The economist John Maynard Keynes famously noted, the ideas of philosophers, both when they are right and when they are wrong, are more powerful than is commonly understood. And practical men who believe themselves to be quite exempt from any intellectual influences are usually the slaves of some defunct philosopher. So I'm going to be spending this session talking to you about an incoherent view on the nature of free will that lurks in the background not just of social science genetics, but large swathes of the social sciences in general. And I'll be talking about how it can negatively impact empirical research in social and behavioral genetics by prejudicing us against genetic explanations for human behavior. This theory drives a culture of recrimination in which accusations of genetic determinism are regularly hurled at and sometimes by social and behavioral geneticists. It also fosters a climate which makes it difficult to conduct genetic research on social science outcomes by encouraging key gatekeepers to place additional barriers in the way of such research. Now, the philosopher Daniel Dennett has a wonderfully evocative quote which captures the key characteristics of this theory I want to talk about. He writes, "There is no more potent source of anxiety about free will than the image of the physical sciences engulfing our every deed, good and bad, in the acid broth of causal explanation, nibbling away at the soul until there is nothing left to praise or blame, to honor, respect, or love." Now, I love this phrase, the acid broth of causal explanation, because it captures the widespread anxiety that to the extent that science shows our behavior is caused by something outside of ourselves, that we cannot be held responsible for it. It also conveys how the threats to human agency are conceived as negatively, nibbling away at the soul until there's nothing left. So Dennett's quote thereby captures the central features of a theory of free will by subtraction, that is pervasive across the behavioral sciences. A theory which suggests that the domain of human agency is whatever is left over after the measured causes of behavioral variation have been accounted for. Once you become sensitized to this theory of free will by subtraction, you begin to see it everywhere. It's implicit in the logic displayed across the social sciences that individuals are victims of circumstance to the extent that their behavior can be predicted by, statistically predicted by variables that are outside of their control, such as the socioeconomic conditions of the household that they were raised in. It's also pervasive in moral and political debates, where individuals are often regarded as less responsible for their socioeconomic outcomes, their health outcomes, or even the crimes they commit based on their childhood circumstances. And we've even seen some research on this kind of topic today. But the conspicuous feature of this free will by subtraction position is that beyond the social determinants of various life outcomes, individuals are assumed to remain responsible agents with power over their own life choices. These statistical intrusions on human agency are treated as limited exceptions to the general rule of human autonomy. But what's left unexplained here is how human agency is supposed to arise at all under a secular materialist framework in which all human behavior is ultimately assumed to have extrinsic causes. Or why scientific knowledge of those extrinsic causes of human behavior should infringe upon our freedom more than causes that have not been identified. So increasingly in social science genetics, this same logic is being extended to genetic influences on human behavior and not just social determinants. So for example, in this paper, Rita Pereira explores how both childhood socioeconomic circumstances and genetic differences in ability can both be used as statistical measures of unfair sources of inequality. For her statistical analysis, she uses the EA3 PGS, but she recognizes that this is an incomplete measure of the full genetic influence on socioeconomic inequalities. Meanwhile, we have this study by Gretz et al. That compares sibling correlations for educational attainment across different countries. And in that, the authors defend the use of this genetically confounded measure of unequal opportunity by suggesting that the genetic advantages that siblings hold in common are another source of undeserved inequality. In fact, they note that by failing to capture the genetic advantages that make siblings different from each other, they're actually underestimating the full effects of family background on inequality. So what these two studies have in common is the idea that there are unfair sources of inequality that are due to external genetic and environmental conditions, as opposed to fair sources of inequality that aren't due to extrinsic causes, but are instead a result of the agent's own actions. So that second clause should give you a moment's pause. In any case, the conclusion that both studies seem to be fumbling towards is that the correlation between monozygotic twins is the best measure of unequal opportunity because it's an omnibus measure that captures all of the genetic influences and all of the shared environmental influences on the trait. In other words, the conclusion they seem to be reaching for is that the non-shared environmental variance, that is, the differences between identical twins reared together, is all that people can be held responsible for. Now, if that idea sounds familiar to you, it's because it was fully worked out by Eric Turkheimer, who has the dubious distinction of giving the world perhaps the most explicit presentation of the theory of free will by subtraction. His clearest formulation of this theory is in his 2011 paper, a comment piece called Genetics and Human Agency, and his theory was recently recapitulated by Paige Harden in her 2021 book, The Genetic Lottery. Turkheimer's theory stands behind Harden's point that your genotype, like the social class of your family, is an accident of birth over which you had no control and a source of luck in your life. In that 2011 paper, Turkheimer explicitly declares that the non-shared environment is free will. And he argues that we can claim this because the non-shared environment quantifies human agency as the variation in a given trait that remains after genetic effects and the effects of the family environment have been taken into account. He even goes on to argue that we can rank different behavioral traits according to their controllability and moral relevance. Weight more than height, personality more than intelligence, depression more than schizophrenia. Now, he doesn't say it in writing, but his table clearly leads us to the reader to draw the implication that we have more control, that we have more control over our sexual orientation than our criminal behavior. So if you think something is starting to sound a bit wrong here, you should trust your instincts. It might be true that the non-shared environmental variance for sexual orientation is quite large, relatively large, but it doesn't mean I can control my sexual orientation or choose the kind of people that I'm sexually attracted to. Something is seriously awry here in how Turkheimer is conceiving the non-shared environment. Let's dig deeper. Why does Turkheimer think that the non-shared environment can measure human agency and the controllability of different traits? on the basis that we haven't identified the specific causes of non-shared environmental differences between siblings. And here he references a review he conducted with Mary Waldron, which found that the cumulative effects of measured shared environmental variables accounted for less than 3% of the variation in behavioral traits. So the logic here is that human beings can't be held responsible for their behavior when we can isolate the specific causes of their behavior. But of course, just because we haven't isolated the specific causes of someone's behavior doesn't mean that behavior wasn't caused. So let's recall how Plomin and Daniels first described the gloomy prospect that we might never isolate the specific causes of non-shared environmental variance. They write that the relevant environmental factors might be unsystematic, idiosyncratic, or serendipitous events. This is to say that the non-shared environment might consist of trillions of sliding doors moments, like catching or missing a particular train, or looking left versus looking right, each of which drives our lives in subtly or dramatically different directions from our siblings, if we have any. So contrary to both Turkheimer and Harden, it's not just genetic and shared environmental influences which are a source of luck in our lives. Most of what we would ordinarily call dumb luck consists of non-shared environmental influences. Moreover, as Harden notes in an important footnote to the genetic lottery, the non-shared environment might substantially consist of random stochastic developmental differences between monozygotic twins, which they can hardly be held responsible for. So this is the basis of a significant intellectual rift between Harden and Turkheimer. It leads Harden to conclude that there's hardly any behavioral variation that isn't explained by extrinsic causes, leaving remarkably little territory for personal responsibility. And by contrast, Turkheimer is eager to carve out a much bigger space for human agency. In fact, Turkheimer goes in completely the opposite direction, extending the gloomy prospect to all sources of behavioral variation and arguing that it is only measured genetic and environmental causes of behavior that infringe our agency under any variance component. So I call this his gloomy prospect theory of free will, as opposed to his non-shared environmental theory of free will. So let's see how Turkheimer's two theories compare when looking at variation in violent criminal offending. This twin study in the Swedish registry reported heritability at 55%, shared environmental influence at 13%, and non-shared environmental influence at 32%. In these pie charts, I've used yellow thatching to shade out the variance that Turkheimer says we aren't responsible for under each of his two theories. So based on his non-shared environmental theory of free will, Swedes are responsible for less than 32% of the variation in their violent offending. Maybe we should cut the sentences of those violent criminals by 2/3. By contrast, when we look at his gloomy prospect theory of free will, we get to claw back control over most of the variation in violent offending, especially that big chunk that was attributed to the heritable variation. I guess we can keep those violent criminals locked up a little bit longer after all. But note something else. If the direct effects of polygenic scores come to explain more of the heritable variation over time, or if more of the shared and non-shared environmental sources of behavioral differences are identified, the degree of agency we have over criminal behavior will shrink accordingly. Now that doesn't sound right, does it? The level of agency we have over a particular trait shouldn't depend on how much we know or don't know about the causes of behavioral variation in that trait. So the philosophical mistake that Turkheimer is making here has been given several names, but the legal scholar Stephen Morse calls it the fundamental psycho-legal error. He explains it thus. Most of the time we possess only imperfect, partial understanding of the causes of behavior. It is important to remember, however, that not possessing knowledge of the complete causal account of a person's behavior does not mean that a complete causal account does not exist. If this is a universally caused or deterministic universe, all phenomena are caused, whether or not we have knowledge of those causes. And he continues, in a causally deterministic universe, all phenomena, including human actions, are fully caused. If causation were per se an excusing condition, no one could ever be responsible for anything. Thus, causation cannot be an excusing condition in law and morals, both of which hold some people responsible and excuse others. Essentially, in the academic discourse on free will, it's understood that you basically have two main options open to you. Either you believe that extrinsic causation of behavior is compatible with free will and that personal responsibility exists, or you believe that extrinsic causation of behavior is incompatible with free will and that personal responsibility does not exist. The first category are called compatibilists, Second category are called hard incompatibilists. There is a third option, which argues that human behavior is only partially due to extrinsic causes, which is called libertarian incompatibilism. But that's essentially a supernatural and dualistic view of human agency, which posits that human beings have an immaterial soul and that every time we make a decision, a miracle occurs that allows us to break the laws of causation that govern the material world. It should go without saying that allowing for supernatural agent causation in our scientific conception of human behavior is a pretty unusual way to go about the secular enterprise of conducting behavioral science. Turkheimer's notion that scientifically unexplained variation in behavior is a yardstick of human agency perfectly maps on to this supernatural theory of free will, because it would suggest that the reason we haven't identified some of the causes of our behavior is because there are none, because some of our behavior is miraculously self-caused. There's just one problem. Turkheimer explicitly rejects the supernatural worldview and rejects the view that our unexplained behavior is uncaused. Right after he introduces this idea that the non-shared environment is free will, he states, not the kind of metaphysical free will that no one believes in anymore, according to which human souls float free above the mechanistic constraints of the physical world. And in the Turkheimer and Waldron review, he concedes that something about the environment must be causing differences among genetically related siblings reared together. So how does he square this circle? I put it to you that he cannot. Perhaps you've seen this meme before. The joke is you can pick any two in your romantic partner: physical attractiveness, intelligence, or emotional stability. You can't have all three. A similar trilemma confronts us in the free will debate. We can only coherently hold two of the following three positions simultaneously. A, that human agency and personal responsibility exist. B, that all human behavior has extrinsic causes. And C, that human agency is incompatible with extrinsic causation. Compatibilism holds that A and B are true. Hard incompatibilism holds that B and C are true. And libertarian incompatibilism holds that A and C are true. By trying to support all three positions at the same time, Turkheimer places himself in an impossible philosophical bind. He wants to be a libertarian incompatibilist, but he also wants to support a secular materialist worldview in which all human behavior, like all other natural phenomena, has extrinsic secular causes. This drives him to support an indefensible position, the position that extrinsic causes only infringe upon our free will to the extent that we know about them, a kind of see no evil theory of free will. I would talk you through this excellent tweet which displays this logic, but I don't have time. Yeah, that's basically all I have time to discuss today, but I invite you to read the full piece when it comes out. I just want to leave you with one final thought, that genetic determinism is essentially a non-problem in academic philosophy. Most non-religious philosophers agree that all human behavior has extrinsic secular causes, and the question of whether we have responsibility for our behavior doesn't hinge on the question of whether those causes are genetic or environmental. Thank you for your attention.
Thank you. And we have time for one question.
It's a little bit of a comment and a question. So that was a very interesting talk. I going back to my first year of grad school and thinking about, I'm a clinical psychologist, and we pretty much talked about, if we had all the data, we could predict everyone's behavior perfectly, which would, it seems like, align with, two of those three sort of poles that you just described. I am curious if you've seen differences in disciplines in the way that they approach free will and how they sort of deal with this. Again, I'm a clinical psychologist, so to me, I just kind of put it all aside and I say, well, who can I treat and how can I improve outcomes? I don't worry about free will. Like, it's just, it's extrinsic to what I need to deal with. So I'm just curious if that disciplinary approach, you've seen it different in different areas.
Yeah, thanks for the question. I can only say anecdotally, I haven't looked at data on differences across different fields. I think in the biosocial sciences, especially in like neuroscience, you get a lot of hard incompatibilists, and this has been bemoaned by. There's a very prominent compatibilist philosopher, just a very prominent philosopher in the free will debate. Unfortunately, Daniel Dennett has passed; he's no longer with us. But the biggest philosopher in that area is probably a guy called John Martin Fisher at the moment. a lot of you will be aware a book by Robert Sapolsky came out a little while ago called Determined, I think it was. But he was basically pushing this hard incompatibilist view. And John Martin Fisher wrote a review in this prominent philosophical review publication, basically bemoaning the fact that behavioral scientists, and especially neuroscientists, they stop at the question of whether our behavior is extrinsically caused or not, and they automatically leap from all of the piling up evidence that this causes this increment of behavioral variation, that they stop the argument at the question of extrinsic causation and don't even really begin to ask the philosophical questions about whether that impacts your free will or not. So I would anecdotally suggest that neuroscience is probably where you get the highest representation of hard incompatibilists. But I actually think across the behavioral sciences, both varieties of incompatibilism are massively overrepresented compared to the philosophical community. And if I could just skip ahead to a slide just to show you what the ratios are amongst non-religious philosophers. 68% of philosophers are compatibilists that don't believe that extrinsic causation infringes on your free will. I've actually covered up the number there, but it's less than 8% are libertarian incompatibilists, and that is the dominant view that we are hearing from Hardin, Turkheimer, and also Kevin Mitchell gives a variety of libertarian incompatibilism with a quantum spin.
Thank you. And that concludes our session in the afternoon. So we will come back in 15 minutes for our keynote talk. Thank you again to all the speakers, and make sure to talk to them if you have additional questions. So see you around.
So it's a real pleasure to have Dan with us this year. So I went back in my email records. I've known Dan since, turns out, at least 2011. And he presented as a PhD candidate from UNC in an early IGSS meeting many moons ago in 11. And so going back there, so he actually remembered this. I quizzed him a little bit outside. He remembered what he presented, of course. And it was creating a PGS for obesity using GWAS hits back in 2011. I mean, this was, if you look at the program, this was like miles ahead in a real direction to the future of what was coming, because most of the rest of the program was still like doing some twins and definitely some candidate genes and like a lot of other approaches that Dan was really on the forefront of moving us away from. So he's of course, as you all saw in his bio, has had an exceptional career since that time, continuing throughout IGSS years anyway. He's always presented cutting edge work that's moved the field forward, like this GWAS. And then finally, the most important thing, knowing him this long, back when he was a PhD student, I can report that yes, Dan has always been this way. You're going to see this in action about forward-looking thinking, doing frontier research, and really doing by far some of the most impressive presentations we've ever seen on IGSS. So I'm very happy to welcome him as a keynote now to Tag C. Dan, please.
I am really bad at standing in front of a lectern and speaking, so I'm going to knock something over. So I'm just going to stand out here where there's less for me to run into. I was already feeling a little daunted about coming after the incredible series of talks we just had, but now, Jason, you've made it worse. So I'll try to live up to that introduction. And also just to say, it's great to be back at this meeting, which I'm thinking of as a continuation of the Integrating Genetics and Social Science meeting that happened at Boulder for all those years. I've been away for, 3, 4, 5, I don't know how long it's been, but it's been a while. And it's great to feel the intellectual energy and the excitement and the interesting questions that we get to ponder while we sit in the audience of these fabulous talks. So thank you for having me back. Okay. My title, I think, in the program is Quantification of Biological Aging. This is the title I tend to use when I speak to social science audiences. I'm going to do both of those things, and I'm going to tell you what geroscience means pretty quickly, so you'll be able to make a little bit more sense of where this is going. But this is a program of research that for me started back in 2015 when I was still spending most of my time thinking about genetic differences between people and how they influenced how they grew up and developed and ultimately their risk for disease. And this parallel program of research was beginning to percolate alongside it, trying to understand how people's bodies fell apart as they grew older with the goal of crystallizing better dependent variables to study the ways in which the experiences we have across life, in particular from the early days, shape our health and well-being across the lifespan. So I have some disclosures. The important one is the third one. I'm going to talk a lot about a measure that you've heard about already today. So thanks to Kyle and others for introducing this Dunedin-Pace construct. But it is a piece of intellectual property for Duke and the University of Otago. And so I do get royalties when the company True Diagnostics sells it to whoever it is that buys these kinds of things. This is the outline. I'm going to talk about geroscience. I'm going to talk about measuring the thing that we want to measure in geroscience. And then I'll finish up with this sort of population application. I actually think this order might be wrong slightly at the end, given what I did with the slides as I was preparing for this. But you get the idea. So this is where we begin. Aging is a thing we all experience as the passage of time. And as many of you who come from departments in the social sciences know, it's a process of personal and psychological growth and development. There are capacities that we gain as we grow older. For much of the lifespan, we increase our vocabularies, but we also gain in wisdom, perhaps in generativity. Our equanimity with the universe improves. This is especially important for those of us who are NIH-funded researchers in this moment. Aging is really an asset in managing the anxieties of the present moment. But from a biological standpoint, aging is mostly about decay and deterioration. And what you see on the slide are sort of two visions of this. One, Luigi Ferrucci's sort of upward sloping aging line representing the interplay between the forces of resilience and the forces of I think the way to think about this is just entropy. So, we're here at a genetics meeting. You can think about DNA as a sort of innovation to organize matter in the universe. It is the enemy of entropy. The genetic program assembles reproductively viable agents from matter. And aging is the other side of that coin. It's the universe trying to tear it apart. And eventually, always, at least so far, unless you're Hydra, the universe wins. Okay, so that's what happens. And on the other side of this slide, what we have is sort of the sum total of maybe 20, 30 years of research into the basic biology of aging, which has gotten under the hood of that process of deterioration and identified a set of characteristic molecular alterations that appear to mediate that age-dependent loss of integrity and resilience capacity that occurs in our cells, tissues, and organs and ultimately makes us vulnerable to disease and disables us and eventually kills us. And these include things that you're familiar with, like telomere attrition, although not what we measure from a blood sample in a human. That's not something you should spend your time studying unless you're an immunologist or Abraham Aviv. And you're studying heart disease, perhaps. Tremendously important within a cell, I don't think interpretable in human blood measurements, with some exceptions. But anyway, you have the loss of proteostasis, so proteins do a lot of work. We're hearing a lot about them in genetics now because genes encode proteins and they are the drug targets that we're often after. But they are amino acid origami, and as we get older, we get less good at folding that origami and less good at identifying the misfolded origami and breaking it down and cleaning it up and getting rid of it. Mitochondrial dysfunctions, we've all heard about these things as the powerhouses of the cell. They do a bunch of other stuff, but as we get older, they get less good at doing all of their jobs. They produce less energy, among other things. Our stem cells get exhausted. Our DNA becomes riddled with errors. Our machinery for recognizing and repairing DNA damage breaks down. Not, as Evan Charney thinks, causing somatic mutation, which is actually the true cause of everything. And anyway, maybe Evan Charney is not somebody we worry about in genetics anymore. We've all moved on. Okay, but anyway, the point here is that this thing that was regarded by biology as unsystematic and uninterpretable for much of the second half of the 20th century, has emerged in the 21st century as a process that we can study. We can molecularly dissect. We can genetically manipulate animals to evade some of these changes. We can deliver behavioral and drug interventions that slow or reverse some of these changes. We can act on this thing that we can now think of as a biological process and not just the error term in development. This has given rise to something called the geroscience hypothesis. So this, I trace this back to a 2014 paper in Cell, but there's actually, I think, a guy out in California who coined the term maybe a year or two earlier. But geroscience is the hypothesis that essentially we have this set of molecular changes that occurs within and between our cells as we get older. Those undermine the integrity and resilience capacity of tissues and organs, making us vulnerable to a loss of function and ultimately the onset of disease and death as we get older. And critically, oh man, I have another slide usually there in which there's like a lightning bolt that comes down and intersects the cell and blocks that red arrow from going forward. But the point is that if this causal model in the cartoon is right, then by intervening to slow or reverse those cellular level changes, we can delay or prevent aging-related disease, and not one at a time in the way of conventional medicine, but all at once with this kind of a silver bullet to build more health span. Okay. So how does that get us where we're going? So this is sort of the model that geroscience and aging biology are suggesting. And in this cartoon, you've got the blue region at the bottom. Those are the healthy years of life. And the red region at the top, that demarcates life that is ill. And you've got time running along the x-axis. Here it's labeled midlife and later life. You could think of this as early life and later life. It's sort of arbitrary how we're marking that. But there are two people in this cartoon. One person is aging rapidly, the red line, and the other person is aging more slowly in the blue line. And the difference between them is just when they transit that border between health span and the period of life lived with disease. And so if that's what the world looks like, there are intervenable states that occur well in advance of the clinical appearance of disease. And we might wish to intervene, in this case, with these folks before the person who's going to age rapidly has really progressed down that path. And geroscience has suggested some interventions that could, in fact, accomplish this. I think I probably don't have the slide that lists those candidate interventions, but they include things like, and I'll come back to this, calorie restriction, so the reduction in total dietary intake. We've known for about 100 years, slows aging in laboratory animals. There are drugs, rapamycin is one we give people today to prevent rejection of organ transplants, but it suppresses the immune system and affects a range of biological signaling in ways that substantially prolong the lifespan of the laboratory mouse and also have salutary effects on other organisms. There are senolytic drugs which target and clear senescent cells from diseased tissues and can do things like improve wound healing, but there's the possibility that systematic senolytic therapy may also be able to preserve health span. But to the extent that we might imagine running trials of these therapies in humans, the duration required to follow up an intervention occurring in young adulthood or midlife to delay the onset of age-dependent disease is decades, and nobody has that kind of time or that kind of money. So what we're after are biomarkers that can function as surrogate endpoints in the context of these trials that can quantify the impact of the intervention that we're attempting to translate from the animal into the human over timescales of months or perhaps a year or two, so that we can know whether we're on the right track to actually launch the full-scale trial that's going to give this drug to a lot of people and follow them for a long period of time. Okay, so how do we do that? Historically, the way we did that kind of thing was we would pull molecules out of basic science research into free-living humans. This is where telomeres came from. This was a biological clock for the cell that we thought could function as a biological clock for the person. And lo and behold, when we measured telomeres in human blood, we found that they tended to be shorter in older people than younger people. And that was really exciting. We thought we had a biomarker of aging. Except that when we follow people over time, their telomeres didn't reliably get shorter, and then it turned out we had huge measurement problems with telomeres, and then it turned out that the telomere length that we observed in blood was a function of a very large number of things, only some of which were the progression of biological aging. And so while they were related to aging biology, as they were available to us as researchers, they were not the readout we were looking for. And the same thing is true of molecules like Clotho, P16, which you can measure in lymphocytes if you can get a hold of lymphocytes, and it's kind of interesting, but those are not readily available in free-living humans. And so we've moved away from this, what I would think of as a hypothesis-driven approach to quantifying the biological process of aging in people, and toward a data-driven approach, where we measure as much as we can about the person, and then we examine how that information relates to some outcome or process that we think represents aging, and from that derive a metric. And that metric then we take forward and establish as a predictor of health span and lifespan, and I'm going to talk about some examples of that. But the idea of this data-driven approach, that we can sort of hack the biology of aging by measuring a lot of biology and seeing how it changes over time, is hypothesized to give us metrics that are variable in young people before the onset of disease. We need that if we're going to use them as clinical trial endpoints. They precede disease and predict it, so they can give us a preclinical assessment of risk. And ideally, they're sensitive to preventive intervention. And that's something that we don't get out of metrics like frailty, which is a very successful means of quantifying differences in biological aging in older people, but which is either invariant in younger people or tells us about problems that we're unlikely to be able to unwind. Okay, so again, I said I got into this business about 10 years ago, and this was the paper working with Terry Moffat and Avshalom Caspi, lab that Kyle came out of as well. We assembled data from the Dunedin longitudinal study. These are 1,000 people all born in the same city between 1972 and 1973 that quantified a bunch of different organ functions at 3 time points, when people were 26, 32, and 38. We modeled how fast those things changed. We composited the individual rates of change for those 18 biomarkers into a single number, which was scaled to represent the change over time they were experiencing relative to the cohort mean. And that gave us this thing we called the pace of aging. That mapped onto this theory in geroscience that if aging is this gradual and progressive decline in system integrity, then we could back that out by watching decline in a bunch of different systems. What was common about those systems would be this thing that we were calling aging. And in fact, the pace of aging performed like we wanted it to. It predicted differences between people in their functional capacities, in their cognitive functions, in how old they looked. But it was absolutely useless from a geroscience perspective, because the whole point here was to get a biomarker that we could measure over months or years, not follow somebody for over a decade in the hothouse of a clinic-based assessment. And so that's where the genomics part of this comes in. So this is DNA methylation. You've heard a lot about it. These are chemical tags on our nucleotide sequence. In biology class, you may have been taught they function kind of like a dimmer switch for gene expression. It turns out they don't. In fact, nobody can really decide what DNA methylation does, but like everything else in the genome, we can be confident it's lots of stuff. But as we study it, it's not so much, we're not studying it as a mechanistic piece of genome biology. We're studying it as a repository of information about the health of the individual and their surrounding environment. And the reason we're studying DNA methylation is because Steve Horvath in 2013, after many years of struggle, managed to publish a paper that kind of blew the roof off of the field of the biomarkers of aging and gerontology more generally, showing that he could predict how old you were with an error of just a couple of years from the DNA methylation state of virtually any cell in your body, except for sperm. They were useless. But this was really a transformative observation. And it invited the interpretation, here crystallized in Bernice Benayoun's cartoon, that we had this measurement of biological aging. It changed across the whole lifespan. It was variable between people. And the hypothesis was that the prediction error in this DNA methylation algorithm that Steve had derived represented that thing that we cared about, that biological process of aging that was disabling and killing us. Unfortunately, it didn't. In an ocean of empirical research at this point, it's clear that the prediction error in this epigenetic clock that Steve first introduced, and in others that have been devised to measure differences between people in their chronological age. So the clock is developed by comparing older people and younger people and then building a parametric model of those differences into a prediction equation. they deliver weak and inconsistent prediction of morbidity and mortality in humans. And Steve, at this point, if you see him give a talk, will say, like, yeah, you shouldn't really use the clock for this purpose. It's not what it's for. It is, nevertheless, a fascinating piece of biotechnology. My blood cells are like maybe 3 weeks old, but they all know how old I am. That's very interesting. It's just not the endpoint for a geroscience clinical trial. And there are a couple of reasons that, you know, we're population scientists, or I moonlight as one from time to time anyway, that you guys are population scientists, that we know that this approach of comparing old people and young people is actually not such a great plan if what we want to do is back out the biology of aging. And the first reason is this thing on the slide. That's Abraham Wald. He was also at Columbia quite a long time ago, and he had the task of figuring out where they should put more armor on the bombers flying missions over Germany. The data they were given looked like the cartoon up there, and those are the bullet holes on the planes that came back. And Abraham said, put the armor where the bullet holes aren't. And the idea here is that it may be the case that Steve's CPG sites are a lot like Abraham Wald's bullet holes, in the sense they are the survivable insults. They are signs of resilience to an aging process, not the succumbing to it. And in addition, we have the problem that, as illustrated in this familiar survival plot below, the older people that we sample into our studies themselves represent examples of successful aging. If you're 75, maybe not today, but certainly when the data in Steve's study were generated, half the babies born in the same year as you were already dead, mostly from age-related causes. And so you had survived those causes. You were a bad model of the bad things that happen to people as they get older. The other reason this first generation of epigenetic clocks might not work so well is the mistaking of what we would call cohort effects for the biology of aging. So in a cross-section of people who vary in their age, we were also looking at differences in the timing of birth, and we know that history has introduced a range of exposures that may embed in our epigenomes, or some of you may not know, but Kelly, for example, is familiar with the ways in which environments embed in our epigenomes. And those could be mistaken for the signs of age in a design like the one Steve used to build his original clock. Okay, so to their credit, the researchers confronting the empirical shortfalls of their tool went back to the drawing board and led by Steve's then postdoc, Morgan Levine, who came out of Eileen Crimmins' lab, so there's a population science tie-in, attempted to overcome in particular the survival bias confound by replacing age as the dependent variable in their machine learning analysis with survival time. And so, you know, they sort of flipped the model around. They said your biological age is the age at which your mortality risk is normal. They built a DNA methylation prediction of that thing, and the deviation from the expected mortality risk prediction turned out to be a much better marker of your risk for disease, disability, and death, and remains so. But there are still limitations. In particular, that we don't know when the variation encoded in this so-called second generation of epigenetic clocks is established. So we have a DNA methylation prediction of survival that reflects a lifetime of accumulated exposure. We know from basic science that the methylome is disproportionately affected by events at the very beginning of life. So it's possible that what we're mining in the epigenetic information is enriched for these early life exposures or causes that may not be amenable to interventions that modify the biology of aging. And in addition, the clocks themselves are optimized to summarize a lifetime of accumulated aging-related change, not the very short period of life when a trial might intervene to modify that process. So they just may not be properly tuned to detect the intervention effects we're after. And so this is where we saw an opportunity and also a template to take this pace of aging construct that we had developed in a cohort study and distill it into a blood test that could actually be useful in the context of a randomized trial. And so the design here is DNA methylation measured at the end of follow-up in the longitudinal study is used to model the average rate of change across a panel of indicators, with the result being an algorithm that reads out a value with an expectation of 1 for midlife adults. That means 1 year of biological change per year of time. Values above 1 indicate faster aging. Values below 1 indicate slower aging. So it's just a ratio of biological change to calendar time is a way of thinking about the pace of aging over the recent past. And the idea is that because these people were only 45 at the end of the follow-up period, we're not worried about survival bias. They were all born in the same year, so we're not worried about cohort effects. And we're modeling changes over the recent past so we know when the variation has occurred. These graphs show evidence that the Dunedin-Pace clock, this epigenetic clock that we built in this alternative way, is as good at predicting health span and lifespan as the mortality prediction clocks. And you've sort of already seen this work from Kyle. These are data on cardiovascular disease incidence, stroke incidence, and dementia incidence in the Framingham Heart Study. The y-axis is a hazard ratio, and on the x-axis are different clocks, with Dunedin-Pace in gold on your far left, Grim age is in navy blue on your far right, and the maroon ones in the middle that are mostly non-significant are the first generation clocks. You see mortality effect sizes in two different studies on the other side of the slide. Again, Dunedin-Pace is sort of keeping up with everything except for Grim age in Framingham, where Framingham was developed as a predictor of the dependent variable in that particular graph. So, but Grim age really is very good at predicting mortality. This is disability, if you're more inclined to think about disability than disease. The point here is just the thing predicts bad stuff that we care about happening. There's the slide I was looking for. So the question is, all right, so we built this mousetrap. Does it actually do a better job of trapping mice? It's comparably predictive, but is it responsive? And so the setting we use to test that response is the calorie trial. Calorie is a randomized controlled trial of caloric restriction in healthy non-obese humans. The reason we're studying caloric restriction is, in my case, partly because of data availability, but in the case of the people running the trial in the first place, because we had 100 years of evidence that it slowed aging in yeast, worms, flies, mice, and rhesus monkeys. At the very least, it prevents the onset of disease. The jury's still out on whether it keeps them alive longer. So NIA ran this trial with partners at WashU, Tufts, and Pennington in Louisiana, and then the coordinating center was at Duke. The goal was 25% caloric restriction, which if you're a mouse researcher doesn't feel like much. They do as much as 40%, but if you're a human, it's a whole lot. But it didn't work out quite as cleanly as they had hoped. The bar chart here, the box plot is showing you estimated caloric restriction in the control group, that's the left side bars, and then the treatment group, that's the right side bars. What you can see is that there's a lot of variability in the control group. These people all did sign up for a caloric restriction study. But it's at least centered on zero. In the treatment group, they got on average to about half of the prescribed dose. So this is really a study of 12% CR, not 25% CR. Nevertheless, the intervention induced substantial and sustained weight loss. That's what the graph on the right-hand side is showing you. About 8 kilograms on average over the first 6 months of the trial, maintained then through the remaining 18 months. And we have some evidence that the thing had positive effects. Now, the pre-specified endpoint in calorie, the primary outcome of the trial, was something called resting metabolic rate. And resting metabolic rate was not durably altered by the intervention. There was a significant difference between treatment and control at 12 months, but not at 24 months. However, there are a range of observations that have been made of calorie suggesting that it did do good things for people. Biomarkers of cardiometabolic disease were dramatically reduced without harming organ size or muscle function. There is maybe some evidence of bone loss that is continuing to be examined. And there are some other studies that suggest that perhaps there's thymic rejuvenation occurring in some people. This is based on gene expression in adipose tissue, so it's extrapolated. But downregulation of systemic inflammation is observed across multiple tissues. We see that in muscle, we see it in fat, we see it in blood. And in a study of a subset of the participants at the Pennington site, where they put people in a metabolic chamber rather than using another method to measure the endpoint, they actually did find that maybe it did slow resting metabolic rate in the treatment group. So some evidence that maybe calorie did its thing. When we use blood chemistry algorithms to estimate biological age, so these are approaches that take a bunch of the analytes that your doctor would measure at your annual physical, and we use them to model age differences between people or mortality differences between people, we find that calories seem to improve those things. So that was a sign in the right direction. And on the strength of this evidence, we generated a multi-omics database for the trial. So gene expression in muscle and adipose tissue, DNA methylation in those tissues and blood. There are a variety of other data, and if you're interested in this kind of thing, you should go to the Aging Research Biobank. They're all available. But this is what we found when we looked at the epigenetic clocks. So what I'm showing you here is change from baseline at baseline when change is 0 by construction, 12 months and 24 months. In the caloric restriction group in red and the control group in blue, AL stands for ad libitum because in caloric restriction, that's what we call the control group. The clocks tick, right? They go up, but they just don't go up differently in treatment and control. That's what you're seeing in the first two panels for phenoage and grim age. These are mortality prediction clocks. I'm not showing you the data for the age prediction clocks. They look just like that. Whereas in the case of Dunedin pace, we see a reduction in the pace of aging by 12 months, which persists through 24 months. There's a slight non-significant increase in the control group. We can talk about why that might be happening, but this is what we see. At the bottom, you see the individual participant data. It's noisy as hell. So there's a lot of individual variability in response to the intervention that's largely not explained by the dose of caloric restriction they achieved, although if we do dose response analysis, we do see a larger effect in Dunedin pace in response to caloric restriction, but not in the case of the other clocks. So this is good. This is what we wanted to see, but of course one wonders why. And I've offered you an argument for why in the lead up here. I've suggested that it's because the Dunedin pace clock is designed to measure changes occurring over the recent past, whereas the mortality prediction clocks may be better designed to summarize a lifetime of accumulated risk. But that's just me saying that. So how would we evaluate this hypothesis? Well, there's a couple of observations that have been made in other studies, but in the interest of time, let me just get to what we did about this. So we would like to have another metric developed using the same method that we are arguing is better at doing this. And it would be even better if we could do that in one of the same data sets that were used to derive the clocks that didn't respond, but that we know are predictive of health span and lifespan. So that's what we did. These are data from the Framingham Heart Study. That's Kaylin Ryan and Will Morella. Kaylin is a research scientist in my group, and Will is a programmer. These are the data from the Framingham Heart Study offspring cohort, the same data that Steve used to build GrimAge, not these data. These are the biomarkers and when they were measured. So unlike the Dunedin study, which had helpfully measured everything in everybody at the same time, 3 times in a row or 4 times in a row in the development of Dunedin pace, Framingham didn't do that. But we nevertheless attempted to model a pace of aging from this sparse matrix of biomarker data. And what we see, what I'm showing you here is if we standardize the biomarkers in Framingham as we did in the Dunedin study, we get this same pattern of upward sloping change. Things are coded so that values that are associated with aging are higher. So for example, lung function is in these data. Lung function declines with aging. It's been reverse scored. But what we see is this nice pattern of aging in the physiological data, and we can model those individual differences in the physiological data, in the change in the physiological data, in this case using a hierarchical model which is simultaneously modeling change across the 17 biomarkers in the data and estimating the shared and biomarker-specific components of the slope for each person in the data set. And so we take the shared component of the slope of those 17 biomarkers for each person as their pace of aging. And unsurprisingly, since all the stuff we're studying is health relevant, people with faster pace of aging die a lot faster than people with slow pace of aging. I think these are just tertiles of participants. We can show that same graph for those outcomes I showed you before. So it looks just like this for heart disease, for stroke. With dementia, the lines are closer together, but they still separate in precisely the same way. But that wasn't really the point. The point was to build another epigenetic clock along the same method as Dunedin pace. And so that's what we did. We used the same set of CPG sites to train the clock. I didn't talk about this before, but Dunedin pace is engineered to have high test-retest reliability by searching a pre-specified subset of CPG sites that we know have pretty decent measurement properties. There are more elegant ways of solving that problem, but for now we're just doing this sort of brute force method. We can then fit a model, and we do 2 versions of this because the GrimAge, for those of you who are not familiar with this thing, is not just a DNA methylation construct. It's actually DNA methylation plus age and sex that are known from the participant because that's at least the information we would have about most humans that we would want to measure. And Steve was interested in keeping the DNA methylation information about age that he had already discovered wasn't so good at telling him about health out of his new clock. So we built one like that, and then we built one like we built the original Dunedin pace, which is pure CPG data. They're both pretty highly correlated with Dunedin pace, maybe not as perfect as you'd like. If the numbers are faint, they're both in the mid 0.6s, the correlations with Dunedin pace, and they're correlated with each other at 0.9 or something. And those are the age residuals. But what really matters is, does it predict health span and lifespan? And what I'm showing you here, these are data from the Women's Health Initiative, 1800, mostly older women. And what you can see is that these new algorithms in blue have comparable effect sizes to Dunedin pace in purple, and then the second generation clocks in red. So they're as good at predicting health span and lifespan. Those are the same results in the US Health and Retirement Study. They're probably a little hard to see, but again, the point is that they're pretty much as good as the competition. Although again, note grim age in the HRS is a real outlier there. It's killing it with mortality prediction. There's no pun intended. And this is what the data in calorie look like. So here, we've residualized all the clocks on chronological age before graphing them, which is why the clocks at the bottom, the pheno age and the grim age, are not going up anymore. This is how Steve prefers to see the data, so that's why I did it. But what you see is that these two versions of Framingham pace, the version that has age and sex embedded in the modeling, which we're calling A, and the version that's pure CPGs, which we're calling B, show the same pattern of response to intervention as Dunedin pace. They decline over the first 12 months of the trial, and that decline is maintained through the second 12 months of the trial. These are the effect sizes. So you can see they're negative for all of the pace clocks, and they're not in the case of the second generation clocks. So that's my empirical argument that this method is actually a better mousetrap. So where I'm leaving you is that, and maybe I haven't really given you the intuition for some of the later points, but I think some of the earlier talks actually have, that these quantifications of biological aging are potentially transformative, certainly for biomedical development of therapies to intervene on aging, but in fact, I would argue even more so for population science, which historically has depended on observations of lifespan to understand differences in the aging of populations, or at the very least, observations of the incidence of disability and chronic disease in older people. So it's always depended on observations of people who it's too late for us to really help. And with the advent of technologies that can quantify differences in aging in people who are much younger, we have the opportunity to observe these population processes in individuals with whom we may still be able to intervene. They're going to be useful in risk stratification. They're going to be useful in population surveillance. Someday, they may even be useful in precision medicine. Labels are what machine learning people call the dependent variables in their regression. And I hope that, for those of you who are users of epigenetic clocks and will be users of the successors to epigenetic clocks, which will be developed from various other ohms or multi-omic data, that what matters may not be most the depth of the molecular data that we're putting into the model or its variety, or the sophistication of the machine learning or AI method we use to fit those molecular data to some phenotype, but the phenotype itself is going to be really consequential for the utility of the algorithm we develop. And so pace of aging was premised on the notion that aging is about change, and I would argue that there are a wide range of change phenotypes one can envision beyond the one that we've developed, but those should really be our focus as we begin the further development of tools to quantify what will be modifiable features of human aging. With the caveat that ultimately, to know whether these things really work, we need sort of the third piece of the puzzle. So it's fine to observe that we have a biomarker that predicts health span and that it responds to an intervention, but until we can show that that biomarker mediates the health span response to an intervention, we don't have something that we can use as a true surrogate endpoint in clinical trials. So we are working on that. The FAST initiative at the American Federation for Aging Research, which I'm leading with Nir Barzilai and Madi Mokri, is attempting to acquire samples from completed clinical trials of drugs that we believe have impacts on aging biology and that have observed follow-up to clinical endpoints. So we can do omics on these samples, identify mediators of treatment effects in these trials, and then take those mediators into other studies to establish the extent to which they predict health span and lifespan and port across drug trials. Ultimately, the vision of FAST is that by intersecting the molecular responses to multiple different drugs that affect aging biology, we can identify their shared signal as a more robust biomarker of aging. So far, We've got one trial in the bag of an SGLT2 inhibitor called dapagliflozin. But hopefully, we will expand this dramatically with support from the ARPA-H Prosper initiative, and you'll see a lot more of this soon. I think, again, this point has already been made by others. In geroscience, the theory of like where does aging come from is the universe. Aging is an outcome of processes of life. Our cells do things in order to keep us alive. Doing those things induces molecular damage to other cells. The accumulation of that molecular damage gives rise to aging. But of course, everybody in this room knows that there's a whole bunch of other stuff outside the person that also impinges on the functioning of their cells, either directly as an environmental toxicants that enter the body, and you'll hear more about those tomorrow from Kelly, as well as via psychological stress processes that you've heard about some today and will no doubt continue to hear about. These are some observations we've made. Sorry, I mean, I feel like you guys have seen a lot of sort of social exposure to epigenetic clock stuff. I don't need to show you more of it, but we've done some of it. One of the things that I think was not maybe clarified earlier today is that even though these epigenetic clocks that we're working with were generally developed in populations of exclusively white European descent people, either white people in Dunedin, New Zealand in the case of Dunedin Pace, white people in Framingham, Massachusetts in the case of Grimage or Framingham Pace, their effects appear to be parallel in other ancestry populations. And in fact, Dunedin Pace and Grimage have now been studied in populations all around the world, and there's robust evidence that they are consistent predictors of health span and lifespan across different genetic ancestry backgrounds. That's what we were doing in that paper. This was sort of another version of social correlations with epigenetic clocks with sibling differences and social mobility as additional, guards against confounds. But other people, I think, including Lauren, have done a lot of work on this subject, and there are many elegant examples. My favorite is Lauren's recent paper in which they used the variation in the depth of the Great Depression across geographic regions to identify an effect of early life economic adversity, in utero economic adversity, on DNA methylation biomarkers of aging. Where we're going with this next is to do some trials. So we can show that a sort of a gold standard geroscience intervention modifies the pace of aging. What we'd like to know is whether changing the conditions under which people live can have similar effects. So the two trials that are funded so far are My Goals for Employment Success. This is an employment intervention delivered to unemployed people receiving federal assistance with housing in Houston and Baltimore. We're almost at the end of data collection there, and as long as we can get the NIH to give us the final years of money, we're going to do this, and you'll know. The other one with Alison Aiello and Ashu Honda is a cash transfer intervention in Malawi, randomized across villages. And the design here is the comparison of early intervention versus waitlist control. So it's 6 years of cash transfer versus 3 years of cash transfer. It's not an ideal design to test this hypothesis, but we're hoping it can get us moving on the way. And there are actually some really interesting early findings from this trial. You can catch them at upcoming Alzheimer's meetings. That's Peter Munig over there, my collaborator who recently just retired to live his best life in Lisbon, Thailand, and Paris. Finally, for those of you who are HRS users, this paper will come out in Nature Aging this summer. We did the pace of aging analysis that I've described to you in the Dunedin study and the Framingham study using data from the health and retirement study, which are much sparser in terms of biomarker coverage for longitudinal data, but which still can give us a longitudinal change construct that quacks like a duck insofar as we see individual differences between people, we see accelerating change toward the end of life, which is what we expect. And the slides are not well, they're in the backup, so I can show them to you. But the point is, it's quite good at predicting health span and lifespan, better, in fact, than cross-sectional indices of biological age or the pace of aging that we get out of DNA methylation. It's observable on a much larger set of people. So we can do this for about 13,000 people in the HRS. So for those of you who have natural experiments you're interested in investigating in the HRS or other exposures, this may be a useful tool. You can email me about it, and then eventually the HRS will distribute it for us. It's also available in the English Longitudinal Study of Aging, which was our replication sample. Code for Dunedin Pace is on GitHub. Code for the blood chemistry algorithm calculations is also on GitHub if you're so inclined. We are available to help you measure biological aging in your data if that's a thing you are into or need help with. This is my thank you to all the people who've worked on these various projects with me, and the people who have paid for them. And this is my thank you to the organizers of this meeting for inviting me, with an acknowledgement to a few folks who I thought were important to the early development of this conference and who are not here today, but who I also owe a debt to. So thank you all for having me, and hopefully it wasn't too much of a letdown after the very deep thinking of our final session. This is more like a digestif, perhaps.
There is time for questions, but we're going to have to corral Dan over to that mic to answer them for a few minutes. Who has questions for Dan?
I was hoping I was going to get to walk around and put the microphone.
So thank you, Dan, for a great talk. I'm just kind of, I don't work in aging research, but I have kind of 2 points that I want to ask you. So do you see that the future of aging research is moving more towards, let's say, extending lifespans so we can live to, let's say, 150 years of age? However, we're going to be suffering from disease for a longer period of time because, let's say, disease onset happens at age 60 and then we just live with disease for a long time? Or are we trying to, let's say we all live to 90 years of age, but we're able to kind of offset the disease happening, so we're able to live with kind of maybe more years without disability? So kind of two points.
I mean, 20, 30 years ago, longevity was the endpoint everybody cared about because it was the thing that they were measuring in worms, flies, and mice, and they figured everybody wanted more of that. These days, the field is very much explicitly organized around increasing what we call health span, or years of life lived free of disease and disability. It remains an open question whether with one you're going to get the other or not. I think there's this idea, and there are some papers that prominent geroscientists published suggesting a limit to human lifespan, maybe around the 120 range. It'd be awfully convenient if that was true, because then the increases we generate in health span shrink the sick years of life. We get this rectangularization of the life experience. So rectangularization of mortality, obviously, is a population observation. But this would be sort of like we live a long, healthy time, and then we die overnight in our sleep. We don't know that that's what's going to happen. We may just be kicking the can down the road. But in the meantime, I'll take some extra healthy years.
Hey, Dan, thanks for that great talk. Really excited to see the kind of work you're doing in deriving these clocks in these methylation measures of aging in HRS and Framingham. That's kind of what my question was about. So I'd always had the intuition that because Dunedin was so homogeneous, that actually gave you better ability to detect differences in aging. And now, Framingham it's generally similar, but a less homogeneous sample, and now HRS too. But you seem to suggest that it's not, these other clocks derived in these sort of more heterogeneous populations are still working just as well. Do you think that there is this sort of value in a very homogeneous population like in Dunedin and being able to port out these algorithms? Or do you think that really it's the longitudinal biomarker piece that actually matters? It's less about having a birth cohort that you've characterized over
That's
time.
a good question. Like, what is the Dunedin study special for reason X or reason Y? And I mean, I think the Dunedin study investigators have long argued that the homogeneity is a strength for this kind of thing, and I would be inclined to agree with them. But the empirical data suggests that it's not necessary. And in fact, another thing we thought was useful about Dunedin was that because they were young, there was no survival bias. We weren't seeing the contamination of the signal by the onset of disease. And so there are various reasons that you might expect Dunedin pace to have some properties that are different. Framingham pace we have not studied as extensively. You've seen pretty much the validation data we've looked at. We've done there's a handful of other analyses in some other cohorts. But from what we're seeing, it appears that what was really important about Dunedin pace was this idea of change. And this is something that aligns with the biologist Rich Miller's argument about what we want out of a biomarker of aging. So Rich is at the University of Michigan and has argued that he has discovered some molecular speedometers of aging in mice. And Rich is a fan of this work, fortunately, because he's a notoriously irascible individual. And I just gave a talk at a meeting. He got up afterwards.
but he was a fan. So I think there's this idea that perhaps what we're going after here with these epigenetic signatures of a change phenotype somewhere in the past is that, like, we call them speedometers, but they may be more accurately described as skid marks, in the sense that, like, this is the consequence of biology changing fast, and that it's not the absolute level we see a person at that is most consequential for their future, but their current rate of change. And that is intuitive and important, but it looks like it has a molecular basis.
Hello, Dan. Thank you for a very exciting speech. So my question came from a different literature. So in the econ literature, we actually try to measure the age in a different way, like for example, using the facial expression and using the machine learning method to back out, let's say based on the eye corners and the number of wrinkles and the skin smoothness, and to see like, and if we see some kind of public figures, they always appeared on TV, then we can kind of see whether under certain kind of pressure, they are kind of aging fast or slow. But on the other hand, of course, such kind of approach doesn't really have any biological evidence. At least they are not exactly linked yet in the econ literature. Big disclaimer. But it seems like this kind of measures using the facial expression could be used as additional phenotypes for you to kind of validate your genetic marks, I guess.
Yeah, so welcome to the party, econs. You're late, as usual. No, but in all seriousness, the reason I say that is because, you know, the computer science people have been after learning age from faces, you know, for maybe 10 years. In fact, in the original Dunedin Pace of Aging paper, the 2015 paper, there's an analysis of facial photographs of study participants rated by undergraduates as looking young or old, and we see that the pace of aging correlates with that metric. And that's true of Dunedin Pace too. There is, I think, you know, you're saying there's no biological evidence for the face age metrics, but actually there are lots of reasons why we might imagine the emergence of wrinkles in the face or the loss of muscle tone underneath the skin or the loss of fat in the skin are signs of biological processes of aging. And I think there's sort of active effort to develop and refine those metrics. There's less putting them together with competing metrics. So like there was just a paper, I think, in Lancet Digital Health about a new face age metric that's good at differentiating survival in cancer patients. So anyway, so I think, yes, to your question, these are absolutely intended as dependent variables for population health studies. That's what excites me, maybe more than anything about them, even if we're building them more purposefully to serve as endpoints in clinical trials. And I think it's going to be valuable to take multidimensional approaches to the measurement of aging that ultimately integrate information like what facial images look like. You know, here we're looking at DNA methylation, but there are proteomic clocks to measure aging. There are metabolomic phenotypes of aging. There are various digital biomarkers of aging that people are pulling out of wearable devices that observe variation in gait and respiration, body temperature. So, yes.
Yeah, thank you for the great talk. We work with the germline DNA variations a lot, which has a really useful feature that it's fixed at conception. It's not really affected by environments. It has a lot of applications. I really appreciate the way you described these DNA methylation-based clocks because it's like it always keeps receipts. It's a cumulative measure of everything you have been through, which really made me think about potential applications. But then you have this speedometer, which confused me because the intuition of that is that if you odometer, always it's a one-way ticket to Paris, right? But speedometer, if you hit the brake, it should come back down. So I guess my question is twofold. First, if you use DNA methylation to build the speedometer, does it actually ever bounce back? So if not, what's your intuition why it cannot, even the pace of it cannot be slowed down?
Well, I mean, like these data are evidence of it bouncing back, right? So the pace of aging declines from baseline to 12 months in response to an intervention here. So the y-axis, again, is a change score. And so when the line dips below 0, what you're seeing is that the average person has experienced a decline in their speedometer value in response to the intervention. That's not a unique property of Dunedin Pace, it turns out. There are sort of good examples like the Do Health trial in Switzerland, which gave omega-3s, vitamin D and exercise to people, reported that omega-3 treatment slowed not just an even pace, but also appeared to slow the rate of increase in the Grimm age and the pheno age as well. Rakov Segal, who's a PhD student, finishing his PhD at Yale and Albert Higgins Chen's lab, has a preprint up that has compiled data from a large number of mostly uncontrolled intervention studies, showing again movement in these clocks in response to intervention, more for Dunedin pace than for the other clocks, but they move too. So modification is possible. And in fact, well, there's another class of observations that I feel like are less heartwarming from the perspective of a clockmaker, observing fluctuation not just in Dunedin pace, which I think is understandable, but in these odometer clocks in response to elective surgery and over the course of pregnancy. So these are very much dynamic constructs. Now, it's important to keep in mind that when I take blood samples from someone a year apart, I'm not looking at methylation states of the same cells at time 1 and time 2. I mean, obviously, I'm not looking at the same cells because they're taken out of blood. But all the cells that were circulating at the time of the first collection are gone, long ago by the time of the second collection. And so this epigenetic memory is being maintained by something other than the cell itself.
So I've got some good news, mostly good news. Dan is not leaving right now. We're about to go on a reception, so we're going to continue this conversation there.